All of you, all of you, all of you. Welcome back, all of you. Good morning, all of you. Now, today we are going to discuss about the molecular basis of inheritance. 100 NEET MCQ sure question. Okay, 100 NEET MCQ sure question. And first of all are welcome to our Saturday classes. If you want more information about my classes, please you joining my WhatsApp channel. I put the link in description box. Okay, guys. So first of all, we are going to the molecular basis of inheritance, and yes, 100 NCRT under 100 NCRT MCQ questions. Okay, ready? 100 MCQ. It's a big chapter and I thought it's the most important chapter for our NEET exam. Okay, ready? Okay, guys, ready? Let us start. And the first of all, look at guys, the first question. It is our first question. Okay. Yeah, it is our first question. The question is come here. DNA. Okay, come on. The first question is DNA. Come on. The DNA molecule, a DNA molecule, the phosphate group is attached to. The phosphate group is attached to which position of the carbon? That's the question. Attached to dash carbon. Come on. The carbon attached to. Okay, the phosphate molecule, the phosphate group is attached to dash carbon. Which is 5 dash. Okay, 5 dash of the sugar, and the nucleotide is carbon is attached to attached to nucleoside, and the sugar residence, it's on nucleotide on the carbon attached to. Okay, yeah, 3 dash of the sugar of the next nucleotide by which bond is a phosphoester bond. It's a phosphoester bond. Which is answer? Which answer? It's a 5 dash, 3 dash. Yeah, option A is the answer. Okay, ready? Option A is the answer. In a DNA molecule, the phosphate group is attached to carbon 5 dash of the sugar, and on the nucleotide and dash carbon, the 3 dash of the sugar, residues of the next nucleotide by dash, which bond? Phosphoester bond. Okay, option A is the answer. Question number 1, option A is the answer. It's a simple question. Okay, simple question. Okay, ready? The next question, guys. Yeah. So simple. The next question is a simple question. Okay, come on. Here guys, look here guys. Phosphate group and the sugar and nitrogen base is called nucleoside. Okay, sugar and nitrogen base is called nucleoside. The nucleoside and phosphate group is called the nucleotide. Okay, nucleotide. Which is option? A is nucleoside. A is nucleoside. A is nucleoside. A is nucleoside. B is nucleotide. Yeah, option C is the answer. So simple question. Okay. So simple question. Okay, ready? Come on, guys. Next question. In DNA strand. Next question. Okay. In DNA strand, the nucleotide are linked together by. In DNA strand, the nucleotide are linked together by. In DNA strand, the nucleotides are linked together by which bond? Nucleotides are linked together by glycosidic bond, no. Peptide bond, no. Phosphodiester bond is the answer. Okay, the two nucleotides are linked together by which bond? The phosphodiester bond. Okay, guys, ready? The next question. First question. What of the following shows you the correct position of the phosphate sugar and the nitrogen based molecule in the given line diagram representing the structure of DNA? Okay, the which is represent DNA? Oh, big question, guys. Okay, just to show once again, once. It's a sugar. Okay, sugar, phosphate, nitrogen base. Is correct? Yeah, chance correct. Sugar, nitrogen base? No. It's a sugar linked with a nitrogen base. No. The next option C is here. Next option C. Option C. Nitrogen base? No. It's a sugar is molecule. No. And the phosphate, no, because option A is correct. Okay, the option A is the correct. No doubts, option A is the correct because the DNA position, sugar is a central. Sugar is central atom. The phosphorus linked with the sugar to sugar. Okay, is a nitrogen base. Okay, ready? Option A is the answer. It's a good. Okay, it's A is the answer. It's a position of DNA. Yeah, good. Oh, Angel Manoj. Oh my goodness. Angel Manoj, okay. Actually, Angel Manoj, it is an English class, okay. I thought you, no problem, okay. The next question. Angel Manoj, okay, next question. Okay, DNA. Okay, the next question. Okay, next question. Angel Manoj, okay, come on. It's the next question. The DNA as acidic substance, okay, DNA as an acidic substance present in the nucleus was first identified by, okay guys, DNA is acidic substance present in the nucleus was first identified. Who is identified? In 1869 is named as, okay, yeah, which is answer? It's okay, okay, come on, which is answer? Come on, which is answer? Yeah, our NCERT does to mention it, like who is that, who is this? The Frederick Miescher, actually the Frederick Miescher is British doctor. He identified some acidic substance in DNA. He called nuclein. Is called nuclein. Okay, which is answer? The Frederick Miescher. Frederick Miescher. Which answer? Frederick Miescher is called nuclein. Okay, the option A is the answer. Okay, option A is the answer. Option A is the answer. Okay, good guys. Frederick Miescher is called nuclein. Okay, option A is the answer. Ready? Good question. Simple question. Pure NCERT question. Okay, next question. The Watson and Crick, 1953, proposed the DNA double helix model and won the Nobel Prize. Their model of DNA was based on. Okay, good question. Yeah, it's a good question. Come on. Based on. The X-ray diffraction data study of DNA done by Wilkes and Franklin? Yeah, is correct. The Griffith base equivalent rule? Yeah, Frederick Griffith. The Griffith, no, it's not Griffith. Chergaff rule. I'm sorry. Chergaff rule. Okay, Chergaff rule state that the adenine equal to thymine and the guanine equal to cytosine. Okay, that's this correct. Okay, sorry, number 2. No. Okay, ready? First of all, is statement 1 is correct, statement 2 is correct. Chergaff rule, base equivalent rule. Okay. The Griffith transformation experiment? No. The Michelson and Stoll experiment? No. Actually, okay guys, the Watson and Crick in 1953 proposed the DNA double helix model on the basis of X-ray diffraction data of Maurice Wilkins and Rosalind Franklin and the rule of Erwin Chergaff. Okay, the two rules actually Watson and Crick together, the two rules are together and proposed DNA because option 1 and 2 is statement is correct. No is no, 3 is no. Yeah, 1 and 2. Okay, option B is answer 1 and 2. Okay, statement 1 and 2 is correct answer. 1 and 2. Okay, B. Yeah, good Angel, good. 6 will be his answer. Okay, 6 will be his answer. Okay, good. Okay, the next question, come on, next question is double strand DNA. Okay, guys, okay. In double strand DNA has 20% cytosine. What will be the percentage of adenine in it? Yes. Come on. Okay, ready? In double strand DNA has 20% cytosine. What will be the percentage of adenine? Yeah, good question. Super question. Angel, come on, Angel. It's a simple question. Yeah, 7C. Good. Because, yeah, 7C. Yeah, adenine equal to, okay, the adenine equal to thymine and guanine equal to cytosine because cytosine is at 20%, then guanine also 20%, and 30%. Okay, option C is the answer. Okay, ready? The next question. In one polynucleotide strand of a DNA molecule, the ratio of adenine and thymine and guanine and cytosine 0.3. What is the percentage of, okay, adenine plus guanine and thymine plus cytosine ratio if the entire DNA molecule, okay, the total molecules. It's one polynucleotide strand of DNA molecule. The ratio of adenine thymine is 0.3. What is the adenine thymine ratio of the entire DNA molecule? Come on, which is answer? Which is answer? Okay, ready? The entire DNA molecules. Which is answer? Come on. Yeah. How to solve this question, guys? The only polynucleotide strand of DNA molecule. The ratio adenine thymine guanine plus cytosine 0.3. Okay. What is the A plus adenine plus guanine by thymine and cytosine entire DNA molecule? Okay, Angel, come on. How to answer? Any possible? Come on, guys, come on. How to solve this question? Guys, come on. Guys, 8A is answer? Oh, 8A is answer? 8A is answer? Okay, just check there. Okay, ready, guys. And just the 8. Okay, guys, look here, guys. The adenine plus thymine plus divided by guanine plus cytosine. Guanine plus cytosine is equal to adenine plus thymine divided by guanine plus cytosine is equal to is 0.3. Okay, 0.3. What is the adenine plus guanine? Adenine plus guanine divided by thymine plus cytosine the ratio. Question, guys, look here. It is a trapped question. It is just a trapped because look here, guys, is entire DNA molecule. The entire DNA molecule means total DNA. The total DNA molecules, the ratio of adenine plus guanine, the ratio of adenine plus guanine divided by thymine plus cytosine. The entire DNA molecule. What's the meaning of entire? The total DNA. Okay, you just calculate the total DNA. The adenine 20 adenine is 20%. Okay, the thymine is 20%. Okay, the guanine is 30%. Guanine is a 30% and the cytosine is a 30%. It is the entire DNA molecule because just divided by. Okay, you can give the answer D option 1 is the answer. The D option 1 because the entire DNA molecule is. Okay, so especially it's a trapped question. It's a good question. The entire DNA molecule adenine plus guanine divided by thymine plus cytosine is a 1. Yeah, because the entire DNA molecule adenine equal to thymine and guanine equal to cytosine because the entire DNA molecule the ratio of adenine plus guanine divided by thymine plus cytosine is equal to ratio is 1. 1 is the answer. Okay, Angel? DC. Okay. Oh, Angel, you okay. I'm sorry, Angel. Okay, ready? Yeah, next. If the sequence Okay, next. If the sequence of the base is one strand of DNA, adenine, okay, thymine, cytosine, adenine, okay, what would be the sequence of base on the complementary strand? Here, simple question. What is complementarity? Complementarity. Yeah. What is complementarity? Yeah. Same question. Super question. It will be okay. Come on. Which is answer? Which is the answer? Okay, what is complementarity? Complementarity, okay, adenine. The opposite is thymine, that is complementarity. Guanine, what is opposite? Cytosine is complementarity. Thymine, what is opposite? Adenine, complementarity. Okay, and the cytosine, opposite. Opposite guanine, it is a complementarity. What is the complementarity? Okay, what is opposite of adenine? Thymine. What is opposite of thymine? Adenine. What is opposite of guanine? Cytosine. What is opposite of cytosine? Guanine. What is opposite of adenine? Thymine. What is opposite of thymine? Adenine. What is opposite of guanine? Cytosine. What is opposite of cytosine? Guanine. What is opposite of adenine? Is a thymine. It is a complementary. Which is answer? Thymine, adenine, cytosine. Thymine, adenine. Okay. Yeah. Thymine, adenine, cytosine. Guanine. Yeah. It is the answer. Okay. Option C is the answer. It is a complementarity. Okay. 9 C. Good. Super angel. Super. 9 C is the answer because that is a complementarity. The next question. The given figure. Okay. The next question, given figure is here. Okay, given figure, they represent the DNA double helix model as proposed by Watson and Crick. Okay, 953. Okay, select the option that show correct measurement of A and B. What is A? Okay, what is A? A is, yeah, A is pitch of DNA. Pitch of DNA. What is B? The distance between two base pair. Okay, distance between two base pair. Okay. The distance between two base pair is, yeah, already our study, the distance between two base pair is 0.34 nanometer or 3.4 angstrom. Okay, the length of pitch of DNA is 3.4 nanometer or 34 angstrom. Okay, yeah, which is answer? Come on, which is answer, guys? Come on. Yeah. Yeah, then which is answer? Okay, give. Yeah, which is answer? Option. Option A is the answer because, yeah, option A is answer because A is 3.4 nanometer. Okay, and B 0.34 nanometer. Option B, A is the answer. Okay, understood? Ready? Okay, ready until here. Good. Ready. The next question, the 11th questions. Now 100 questions. Okay, only one video, just only it's only one part. The complete questions solved. Okay, 100 questions. Okay, it's a simple question. Look at the given figure. Okay. Look here, guys. It's a K. And K is given here. It's a N is given here. Also is M. Is L is here. It is L. And M. Yeah, it is M. Okay. K. Who is K? It is, yeah, it is 5 dash end. K is 5 dash end. Yo, 3 options are 5 dash end. Okay. Who is L? Yeah, it is L. L is hydrogen bond. L is hydrogen bond, triple bond hydrogen bond. The 2 options are given. Okay, hydrogen bond, C or D. Answer C or D. Okay, and M is a adenine or guanine. Is adenine or thymine because it's a double bond. It is a double bond because that is adenine. Yeah, okay, that's okay, equal. It's adenine. Okay, M is adenine. Okay, then N. Who is N? N is ribose sugar or deoxyribose sugar because it is a DNA molecule, and the deoxyribose sugar, option D is the answer. Okay, option D is the answer. Good. Ready. Come on. Option D is the answer. Okay, Angel. I thought it's a simple question. Yeah, Angel is a simple question. Option D is the answer. 11. Option D is the answer. Ready? Go next question. Okay, ready. Next question. The 12th question. Wow, simple. It's a pure NCERT. It's a pure NCERT question. 12th question. Come on. Okay, ready? Come on. The next question. 12th question. The given flow chart represents the flow of genetic information between biomolecules. Identify the process of A, B, C, D and select the correct option. Yeah, simple question, guys. A, B, C, D. What is A? A, what is A? A is, yeah, 11D. 11D. Okay, ready? 11D, okay. Super. 12C, 12C. Oh, 12C, okay. Is a replication, and B is transcription, and C is reverse transcription, and D is translation. Very good. Super question, okay. Very good, very good. Very good, Angel. Angel Manoj. Very good, very good. Next question. The 30th question, 13. Okay, next question, 13. 30th question. The synthesis of DNA from RNA is explained by. The synthesis of DNA from RNA is explained by. Synthesis of DNA from RNA is called translation, no. Reverse transcription, no. Not a replication, is a transcription. Simple question, guys. It's a simple. Transcription is the answer. Okay. Yeah. So D is answer. Transcription is the process of synthesis of RNA from DNA molecule is called. Okay. Good question. Okay. Ready. Simple question. Simple question. It's called a transcription. Okay. Next question. Okay. And the 14. Histone proteins are. Who is histone? Okay. Next question. The histone proteins are. Who is histone? Histone is basic or acidic. Okay. Just imagine, guys. The DNA is acidic molecule. The DNA is an acidic molecule. No, DNA is a negatively charged acidic molecule. DNA is negatively charged acidic molecule, rapid with positively charged basic protein. That statement you okay, this statement is a very, very important. DNA is negatively charged acidic molecule, rapid with positively charged basic protein is called histone. Okay, these statements are very, very important. First of all, okay, guys, the negatively charged acidic molecule is DNA, acidic DNA, negatively charged acidic DNA, rapid with positively charged, the basic protein is called histone. Come on, histone proteins are, so simple. Histone proteins are 14D, yeah. Histone proteins are, option D is answer. Angel? 14B. Yeah, 14B. Yeah. Histone is a basic and positively charged. Option B is the answer. Angel, okay, super, Angel. Super. Okay, ready? It's a simple question. The next question. Okay. Next question. What the given diagram represent? Nucleosome, chromatin, histone complex, both A and B. Yeah, they are represented by. Just confuse it? No, it's a simple question. Yeah, what is it? It is nucleosome. Option A is the answer. Okay? Yeah, option A is the answer. Not a chromatin and not a histone complex. It is chromosome. It is nucleosome. The nucleosome contain histone octamer and 200 base pair DNA. It is 200 base pair DNA. It's called nucleosome. Okay, ready? Okay, ready. The next question, 15, okay, the 16th question. 16th question. Refer the given figure, the nucleosome, and select the option correctly identified the part A, B, C. Okay. Correct part. Who is A, B, C? Who is A? Who is B? Who is C? Okay. Yeah. Who is A, B, C? 15A, okay. Angel, okay, 15A, okay, good. A, B, C, come on. Who is A? Yeah, A is histone octamer, not a DNA. Yeah, histone octamer. Two options are given. Histone octamer. Who is B? Is a DNA. B is DNA. And C, H1 histone. Option C is the answer. It's a simple question. Simple question, guys. Option C is the answer. Simple question. Option C is the answer. Okay. The next question, 70th question. It's a 70th question. The structure of chromatin seen beads on string when viewed under electron microscope are called nucleotide, nucleoside, histone octamer. Sir, ഞാൻ ക്ലാസ്സിലാണ്. ഓൺലൈൻ ലൈവാണ്, അതാ ഞാൻ ഫോൺ ലൈവിലാണ്. ഓക്കെ ശരി സാർ. ഓക്കെ ഓക്കെ okay guys, start. Okay, come on. Yeah guys, come on. Which is answer? Yo, pineal D. Oh yeah, okay pineal D. Okay, angel, good. Okay, the structure of chromatin in beads on string when viewed under electron microscope are called nucleosome, histone octamer, nucleoside, and nucleotide is nucleosome. Ready? Option D is the answer. Okay, option D is the answer. Ready? Good question. The next question, the packaging of chromatin. The next question, the packaging of chromatin at a higher level require additional set of protein that are referred to as, which type of protein needed for packaging of chromatin? Who is protein? It's a pure NCERT question. Histone protein? No. The non-histone chromosomal protein, NHC? That is answer. That is answer because you're NCERT question. Okay. Yes, good. Pineal B. Okay, good. Angel. Okay. Angel is correct answer. Okay, next question. Read the following statements and the select correct option. Loosely packed and okay. Oh yes. Correct options. Okay, good. Loosely packed and lightly strained region chromatin are called heterochromatin. Two type of chromatin, euchromatin and heterochromatin. Okay, nucleus contain two types of chromatin, euchromatin and heterochromatin. Okay, the loosely packed and lightly strained region chromatin are called heterochromatin. The statement are correct? No, because it is euchromatin. The statement are incorrect. Okay, ready? The densely packed and dark stained region chromatin are called euchromatin. Is statement correct? No, the statements are incorrect because it is here. It is the statements are heterochromatin. The next question, a typical nucleosome contain 200 base pair DNA helix. Okay, statements are correct. 1 and 2 are true, but statement no. The statement 1 and 2 are false, but statement 3 are true. Yes, that is answer. Okay, option B is the answer. Yes, good. Angel, good. Super Angel, super. Okay, the next question, select wrong statement about heterochromatin. Okay, select wrong statement about heterochromatin. Okay, it is densely packed. The statement is correct. It's a densely packed. And the stain dark. Yes, statements are correct. It's a dark. Okay, and it is transcriptionally active. It is transcriptionally active. No, it is not a transcriptionally. Euchromatin only transcriptionally active. The statement are false. Okay, the option C is the answer. Okay, yes, Angel, good. Option C. Okay, C is the answer. The next question, next topic, the search for genetic material. Okay, the next area, your NCERT. It's a good question. Okay, is a very important sections. The search for genetic material is very important section. Okay, come on. The purpose of transformation is not affected by which of the following enzyme? The process of transformation is not affected by which of the following enzyme? Okay, actually it is our Griffith experiment. After the Griffith experiment, the search for genetic material DNA, Oswaldo Overy, Colin MacLeod, McCarthy experiment. Okay, come on. The process of transformation is not affected by which of the following enzyme? Okay, DNAs, RNAs, peptidase and lipase. Which is answer? Okay, given. The process of transformation is not affected by which of the following enzyme? Come on. Okay, given. Given. Which is answer? The DNA is affected. Okay, sure. But RNA is not affected. Okay. Because transcription DNA using the enzyme DNA, the transcription not take place. But the RNase enzyme used, transcription take place. Peptidase is used, transcription take place. Lipases is used, transcription take place. Because DNA only. Which is answer? A, no. A, B, C, D, no. Option, look at guys. B, C, D. Okay, B, C, D is used, transcription take place. It's not affected which of the following enzyme? The process of transformation is not affected. Yeah, not affected. BCD is not affected. Okay, DNA is affected because option C is the answer. Is a good question. C is the answer. Okay. Okay. So ready? Understood. So DNA's enzyme is used because transcription is not take place. Okay. Transcription is not take place, but another three enzymes, the peptidase, RNase, lipases used because transcription take place. The mice is killed because not which enzymes are not affected. RNase is not affected, peptidase is lipases. Okay, so good question. Okay, which option? Option C is the answer. Okay, ready. Next question. If the DNA, okay, next question. If the DNA of virus labeled with P32 and protein of the virus is labeled S35, after attacking E. coli cells, which molecules would be present inside the bacteria cells? Okay, who is enter into the bacterial cells? Yeah, who is enter the bacterial cells? P32? P32? Yes. No? S35. Both A and B, no? None of this. Okay, which is answer of P32 because P32 is radiated the DNA. DNA is enter into the E. coli cells because P32 is the answer. Okay, P32 is the answer. Okay, ready? Good question. Ready? Okay. Angel, okay, Angel? Ready. The next question 23. Next question 23. 23. To prove that DNA is, okay. To prove that DNA is a genetic material, which a radioactive isotope are used? Hershey and Martha Chase experiment in 1952. Their experiment, which type of isotope? Simple question, guys. Not a nitrogen. Nitrogen is no. Only P and S. Okay. Yes. P32 and S35. Okay. P32 and S35. Option B is the answer. Okay, ready? Simple question. Option B is the answer. Ready? The next question. So simple. Next. Okay, ready? Okay, Angel? Okay. The RNA is the genetic material in. Yeah, super question. It's a super question. RNA is the genetic material in prokaryotes, eukaryotes, tobacco mosaic virus, and E. coli. Come on. Prokaryotes? No. Eukaryotes? No. E. coli? No. E. coli is DNA. The answer option C. Tobacco mosaic virus. Okay, ready? Yeah, C. Good. Angel, okay. 24C, okay. Good. Ready? The next question. Which of the following? Next question. Okay, next question. Which of the following? Okay. Which of the following criteria should be fulfilled by the molecule act as a genetic material? Who is the best genetic material? The property of the genetic material. That's the question. The property of the genetic material should be able to replicate. Yes. It should be structurally and chemically stable. Yes. It should be able to undergo slow mutation. Yes. It should be able to express itself the form of Mendelian character. Yes, the four is it is your NCRT line. Okay, which is answer? Yeah, answer is option D. Statement 1, statement 2, statement 3, statement 4. That is the correct answer. Option D is the correct answer. Ready? Option D is the correct answer. Ready? The next question. 26th. Select the correct statement out of the following. Okay, next question. Ready? Next question. Question number 26. Ready? Okay, select the correct statement out of the following. Correct statement out of the following. Okay, the both DNA and RNA able mutate. For the DNA and RNA able, yeah, able mutate. Correct statement. Okay, correct. And RNA being unstable, mutate on faster rate. Yeah, is correct. DNA is RNA is very fastly mutated. Okay. RNA show catalytic property. Yeah, that's correct. Ribozymes, example ribosome is a RNA that act as a genetic material. Sorry, that act as a enzyme. The presence of uracil plays thymine is additional stability of to RNA. No. The presence of uracil at the place of thymine is additional. No, it decreases stability. It's a no. It's a no. No, not as answer. Okay. Actually, the most DNA is the most stable because the thymine provide additional stability to DNA. Okay, uracil not stable because additional OH, the additional OH present in the uracil because RNA, DNA is more stable than the RNA. The statements are incorrect. Which options are answer? Okay, select correct statement. 1, 2, 3. 1, 2, 3 are the correct answer option. D is the correct answer. Okay, ready? 26. Okay, Angel. 26 D is the answer. Okay, ready? Good, Angel. Good. The next question is RNA world. The RNA world is the answer. The 27 is RNA world. Which one among the following was the first genetic material? Yeah, it's simple. Who is the first genetic material? No, yes, it's RNA is the answer. RNA is the first genetic material. Simple questions, guys. Simple question. Okay, RNA is the first genetic material. Okay, yeah, ready. It's a simple question. Okay, next question. Okay, simple. Okay, next. Which of the following life process are evolved around RNA? Which of the following life process are evolved around RNA? Oh, good question. Which of the following life process evolved around RNA? It's a metabolism and translation and splicing and all of this. It's a good question. It's a good super question. The following process of, yes, actually our NCERT book and NCERT test book, actually our NCERT test book mentioned the life process evolved from RNA by metabolism, translation, splicing, everything, because all of this. Option D is the answer. Okay, option D is the answer. The life originated from RNA, evolved from RNA because the all process are taken. Okay, metabolism, splicing, translation. Answer all of this are answer. Okay, ready? It's a good question, super question. You just write this question your NCERT test book. Okay, because it's a rare question. Okay, next question. Which of the following statements are incorrect? Okay, which of the following statements are incorrect? Okay, RNA act as a genetic material as well as catalyst. Okay, yeah, statements are correct. Good. The next question. RNA is the more stable than DNA. No. RNA has evolved from DNA. No. RNA evolved from DNA. No. DNA evolved from RNA. The statements are incorrect. Which of the following statements are incorrect? 2 or 3 are incorrect. 1 and 3 no. 2 and 3 yes. Option B is the answer. Option B is the answer. Okay, option B is the answer. Ready? Option B is the answer. It's a simple question. And 30 is a question. 30. Question number 30. Okay. The chemically RNA is a. Okay, guys. Look here, guys. Next question. The chemically RNA is a. Okay. Chemically RNA is. Come on. Chemically RNA is dash reactive and structurally dash stable as compared to RNA. Come on, which is answer? Come on, guys, it's a simple question. Simple. Yeah. Okay, Angel, your correct answer. Okay. Yeah, correct answer. Yes. 29B. 29B. Okay. 29B. Okay. 30. 30. The RNA is more reactive. Okay. RNA is more reactive. Yeah, it's a more reactive, and structurally less stable. Structurally less. Yes, it's a option C is the answer. Okay, ready? Yeah, that is C. Oh, good. That is C is answer. The next question, replication. Oh, yeah, it's a very important section. The replication. Okay, 30. Okay, guys, now go. 30 is answer. The replication. The first experimental proof for semi-conservative of DNA replication was shown in. So, first experimental proof for semi-conservative DNA replication was shown in. Who is the first experiment? Semi-conservative. Streptococcus pneumoniae, Escherichia coli, Neurospora crassa is a VCF Weber. Oh, no. Yeah. Which is answer? Come on, guys. It's a good question. 31 question. Good question. Which is answer? Yeah, 31B. Yes, good. Ready. Yeah, yes, 31B. Escherichia coli. Yeah, that is answer. Good, mister. Yes, ready. Escherichia coli is good answer because pure NCRT. And 32. Which of the following phenomenon? Okay. Which of the following phenomenon was experimentally proved by Messelson and Stahl? Matthew Messelson and Franklin Stahl experiment. Transformation, transudation, semi-conservative DNA replication, and central dogma. Which is answer? Yeah. Which is answer? Transformation? No. Central dogma? No. Semi-conservative DNA replication. Okay. The Matthew Messelson and Franklin Stahl experimentally proved DNA replication is semi-conservative. Option C is the answer. Good, okay? Ready. And the next question. The average rate of, okay, next question. The average rate of polymerization of base pair during replication in E. coli is? Yeah, good question. Actually, our NCERT book mentioned the average rate of polymerization of base pair during the replication in E. coli. In E. coli is how many base pair per second? Per second, 2000 base pair. Okay, NCERT question. Which is answer? Yes, option C is the answer. Yeah, option C is the answer. Okay, ready? It's a good question. 200 base pair per second. The polymerization of DNA, the polymerization of nucleotide during the DNA replication is 2000 base pair per seconds. Okay, ready. The next question. Okay, 34. Question number 34. Did you see? Okay. Then it's select the incorrect statements regarding DNA replication. Incorrect statement regarding DNA replication. Okay. Continuous synthesis occurs in 5-3 direction. Yeah, correct. Incorrect statement. It's a correct statement. Okay, then the discontinuous synthesis fragments are found in 5-3 direction. Discontinuous synthesis fragments are formation in. Yeah, that's correct. Yeah, correct. Continuous process take place 5-3. Discontinuous also 5-3 is correct. Correct. DNA polymerase catalyze polymerization in 5-3 direction. Yes, correct. The DNA polymerase catalyze polymerization in 3 dash. That's incorrect because the enzyme, the three statements are correct. Continuous synthesis occurs in 5-3 direction. Discontinuous synthesis also occurs in 5-3 direction. And DNA polymerase enzyme catalyze polymerization 5-3 direction. Okay, but The DNA polymerase catalyzes polymerization, not a 3-5-. These statements are incorrect. Option D is the answer. Okay, option D is the answer. Good. The next question, 35. Which of the following difference incorrectly between continuous and discontinuous synthesis of DNA? Okay. So very, oh my goodness. So other question is here. Okay. First of all, read this. Continuous synthesis of DNA. It do not require DNA ligase for the growth. Continuous synthesis. Yes, it is do not require DNA ligase for its growth. Yes, its statements are correct. Discontinuous synthesis. DNA ligase is required for joining fragments. Yes. Okay. The statement 1 is correct. Next, the formation of leading strand is slower. No, this formation of leading strand is faster. The formation of lagging strand is rapid. No, the statement B is incorrect. Okay, ready? Incorrect. And statement 3. It is template occurs. Template occurs open in 5-3- direction. Is correct? Correct? No, something problem. Okay, first of all, guys. Okay, this reading, this right here. 3-5-, and DNA synthesize 3- to 5-. No. That picture, I want that picture because it's confusing. So guys. Okay, this area. It's a continuous synthesis. It's a template 3- to 5- direction. Okay, it is continuous synthesis. The template is 3- to 5- direction. Okay. It's template opening. No, the statement is incorrect. 3- to 5- direction. The template opening 5- 3-. Actually, the statements are incorrect. Ready? Okay, ready. And the formation of strand beginning immediately of the beginning of the replication. The formation of strand beginning immediately at the beginning of replication. The formation of strand begin a bit later than that of leading strand. Come on, which is answer? Angel Manoj, 95C. 95C is answer. Come on. 95C is answer. 95C. 2 and 3 only. 2 and 3 only incorrect. Actually, question is what is correct or incorrect? Incorrect statement. Okay. 2 and 3, the statement. Option 4 is correct. Yes, the formation of strand beginning immediately at the beginning of replication. The formation of strand begin at later than. Okay. Option C. Yeah, option C. 2 and 3 are only incorrect, but statement 1 and 4 is correct. Option C. Okay, good, Mr. Angel Manoj. Oh, I appreciate you. Some more difficult question. Okay, good. Option C is answer. Okay, next question. Which of the following figure correctly represent the replication fork formed during DNA replication? Yeah. Super question. Which is answer? Come on. DNA replication. Come on. First of all, guys, 5- to 3-. No, not as answer. Yeah, look guys, it's a 5- to 3-. Correct? 3- is a template. Okay, but problem. No, that's a no. This is no. It's incorrect. The figure. 3 dash, no, that's not answer. Oh guys, yeah, 3 dash to 5 dash, 5 dash to 3 dash, the 5 dash to 3 dash. Okay, yeah, it's this answer. Yeah, B, and this is B's answer. No, no B. Is this answer? Option D is answer. Yeah. Okay, guys. And this is B. B is answer. B or D? This is confusing, guys. Oh, first of all, guys. Yeah. 5 dash to 3 dash and 5 dash to 3 dash. Yes. Which is answer? D. Angel, D is answer? D or C? Sure, D is answer? Okay. Yeah, D is answer. Okay. Because the 5 dash to 3 dash. 5 dash is a lagging strand. 5 dash to 3 dash. B is 3 dash to 5 dash. No. It's option D is the answer. Okay, ready? Okay, good. Ready, next question. Next question, 37. Question number 37. DNA replication take place at dash phase of the cell cycle. Yeah. DNA replication take place dash phase, G1 phase, S phase, G2 phase, M phase. Yeah. DNA replication take place. Yes. SSR, DD. Okay. Come on. Yeah. Which replication take place at a dash phase? G1, no. Yeah, yes means synthetic phase. The synthetic phase take place DNA replication. The DNA duplication take place at S phase of cell cycle. Okay, the option C is the answer. S phase. Yeah, Fatima Selva. Okay, Selva. Ready? Okay. It's good. Ready? Next question. The 38. Refer the given steps of DNA replication. Okay, the first. Come on, which is answer? Which is answer? The exposure of DNA strand, DNA polymerase attach at a ORI site. Oh, actually it is a replication steps, okay? Guys, it is a steps of replication, okay? Ready? Reading very, okay? Carefully reading, okay? The statements are reading very carefully reading. First of all, exposure of DNA strand, the DNA polymerase attached at ORI site, activation of deoxyribonucleotides, chain formation, base pairing, proofreading and DNA repair, because it's steps of replication. Which is first steps? It's a very difficult question. First look at it, guys. First steps. Yeah, ORI is given here. The first of all is ORI. Okay. The DNA polymerase attached at the ORI site is the first step of replication. It is the first step of replication. Okay. The two options are given. Option A and option C are given. Okay. Other is removed. C or D is removed. Okay. The next is 3 or 1. 3 or 1 is answer. Okay. 3. Activation of deoxyribonucleotide and one explosion of DNA strand. Explosion of DNA strand, which is first take place. The first take place is the 3 is first take place because activation of deoxyribonucleotide. The deoxyribonucleotide is activated, then explosion of DNA strand, and number 3, step 3 is here is a 1, and next step 5 is next step. The base pairing is take place, and the next step is for chain formation, and the next step the proofreading and the DNA repair. Option A is the answer. Okay, option A is the answer. Yeah, maybe you think the B is the answer, but actually it's not a B, not a B at all, not B. Yeah, 38 is A is the answer. Yeah, it's a difficulty question. It's a small difficulty question. Okay, just you take the screenshot and after this class you reading your note and your NCERT test book. Okay, you take a screenshot. Okay, ready. The next question, transcription. Yeah, the next area is a transcription. Transcription area. Okay, it's a very important section, guys. Transcription area. So simple. The process of copy genetic information from one strand of DNA to RNA is termed as a simple question. Is a termed as. Yes, answer. Okay, come on, Fatima Silva. Come on. Which is answer? The transcription, the process of copy genetic information from one DNA strand into RNA is termed as, not a replication, not a transcription. Translation, yeah, is a transcription is answer, not a translation and not a reverse transcription. The answer is transcription. Okay, good. It's a simple question. Simple question. Okay, ready. Okay, simple question. Next question. The given flow chart show central dogma. Okay, enzyme used in process of ABCR respectively. Okay, the enzyme DNA, it is DNA replication, it is transcription and it is reverse transcription. The name of enzyme, okay, ABCD. Okay, yeah, ABCD. A is transcription. First of all, A is replication, B is transcription and C is, okay, first, okay guys, A is replication, B is transcription, C is reverse transcription. Which enzymes are used? Come on, which enzymes are used? Come on, which is answer? Replication is used. DNA dependent. Come on. Replication is used. DNA dependent DNA polymerase. Yeah, this is correct. DNA dependent DNA polymerase. This is correct. DNA dependent RNA polymerase. No is no. Option A or B is answer. Okay, next question. Transcription. Transcription used. RNA dependent DNA polymerase. No. DNA dependent RNA polymerase. Yes, is correct. And RNA dependent DNA polymerase. Yes, it's a reverse transcription. Reverse transcription is the process of synthesis of DNA from RNA. Okay, synthesis of DNA from RNA is RNA dependent DNA polymerase. Option B is the answer. Ready? Yeah, for B. Option B is the answer. Okay, good question. Simple question. Yes, 14B. Yes, Angel. Okay. Guys, okay. The next question. Okay, simple question. Next question. The enzyme DNA dependent RNA polymerase catalyze. Okay, next question. The enzyme, come on. The enzyme DNA dependent RNA polymerase catalyze the polymerization reaction in which direction? Yeah. All enzymes are working only one direction, that is 5' to 3' direction. The DNA dependent DNA polymerase, the DNA dependent RNA polymerase is working only one direction, 5' to 3' direction. The enzyme DNA dependent RNA polymerase catalyzes the polymerization reaction in 5' to only 5' to 3' direction. Because 41 option A is the correct answer. Okay, ready? Yeah, so simple. A is answer. Option A is answer. Yeah, this is a very important question for it is sure NEET 2026. It is sure NEET question, guys. Sure NEET question. What would be the base sequencing RNA transit to obtain from the given DNA? Okay, it is a DNA strand is given, is a coding strand and non-coding strand. The question, what base pair of RNA transcript? Yeah, is RNA. Who is RNA? Coding strand is given and non-coding strand is given and it is RNA. Which is RNA? It's a very, very important question. Come on. Which is answer? Which is answer, guys? First of all, it is non-coding strand. Coding strand and non-coding strand. What is non-coding strand? Non-coding strand is template. First of all, you understood the non-coding strand is called a template. Template strand, coding strand. Okay, It is a template strand. Okay, it is a template. Non-coding strand is template strand. Okay, the opposite, like, okay, just a complementarity of the complementarity of template strand is RNA. Okay, it is cytosine, just opposite to guanine. It is guanine, opposite to cytosine. It's a thymine, opposite to adenine. It's adenine, opposite to uracil. Adenine, opposite to uracil. Guanine, opposite to cytosine. Cytosine, opposite to guanine. Cytosine, opposite to guanine. Guanine, opposite to cytosine. Adenine, opposite to thymine. Thymine, opposite to adenine. Cytosine, opposite to guanine. Adenine, opposite to thymine. Not a thymine, it's a uracil. Okay. Okay, thymine opposite. Adenine opposite, uracil. Thymine opposite, adenine, cytosine. Come on, it is RNA. The 5 dash to 3 dash. Come on, which is answer? Which is answer? Guanine, cytosine, adenine. Guanine, cytosine, adenine. Guanine, cytosine, adenine. Two options are given. Guanine, cytosine, adenine. But but a TT, no, not a thymine, Uracil present in the RNA. Option A is answer. Simple question. 42, A is answer. Okay, but one questions are trapped question. The one question are trapped because, okay, because it's a non-coding strand of DNA is template. Okay, coding strand is not a template. It's a very confusing question. I think it's a very confusing question. Okay, you solve one or more question. No, okay, this is a simple question, but it's a very it's a simple but most difficult question. Okay, confusing question. Template and coding and non-coding. First of all, you understood the non-coding strand is called template. Okay, ready? Okay, next question. The next question, come on. If the sequence of base in coding strand of DNA ATCGTG, okay, then the sequence of base in mRNA will be, yeah, simple question. Come on. Fatima Selva, option A is answer. Angel Manoj, option A, good. Okay, come on guys, follow me. Okay, you can crack NEET 2026. Okay, you can crack. You work very hard. Okay, come on. If the sequence of base in coding strand of DNA is a coding strand, first of all, the given strand is coding strand, not a template. The given strand is, first of all, the given strand is coding strand, not a template. Then the sequence of base in mRNA will be, okay, the base in mRNA will be, okay, mRNA. First of all, coding strand is given. The RNA is the same but small change. Okay, first of all given, guys, look here guys. Okay, first of all, it is RNA. It is a coding strand. What's the difference? GG, CC, AA, UUTT. Okay, actually the RNA and coding strand are same, but small change is the thymine instead of uracil. Okay, the coding strand and the RNA are same. Okay, the coding strand and the RNA the similar. The small change, the uracil instead the thymine. Okay, ready? Okay, given. Okay, this question is given here. The given here is coding strand. The question, what is RNA? The RNA coding strand are same. Okay, it's the same. It's the same, and also it is also same. Okay, but thymine instead uracil. Okay, thymine is given. No, the thymine instead uracil. Okay, A. Thymine instead uracil. UUCG. AUG. Option D is the answer. Ready? Option D is the answer. Ready? Good. Ready. Yeah, good. Okay, all of you, I think you can solve. Okay, the next question. If the sequence of base in DNA in 3' to 5' GTG, okay, then the sequence of base in transcript will be, okay, the sequence bases in DNA actually is given here, is a template strand. Okay, the template is given, then the sequence of base in transcript, because question is what is RNA. Okay, ready? Template is given and question is what is RNA. Okay, you write just opposite. G is given, opposite is C. C. Two options are C. C or B is answer. Okay. And C opposite G. Okay. C opposite G. Okay. And T opposite C and G. Okay. C, G. Okay. Ready. Two options are same. C and G. And T opposite A. Two options are A. And next A opposite adenine opposite thymine instead uracil. It's a uracil and it's a thymine. No. It's a no. Option C is the answer. Okay, because option C is the answer. It's a good question. 44B. 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 No. 44C. Because thymine, not a thymine, is a uracil. Okay, it's a very important question because yeah, it is trapped question. Yeah, sure, it is a trapped question, guys. Yeah, it's a trapped question. Okay, the answer 44, option C is the answer. C is the answer. Okay, good question, guys. Okay, go next question. Go next question. C is the answer. Ready? Angel Manoj, okay, you understood? Okay, Fatima Selva, okay? Simple question? Okay. Okay, ready? Next question. Okay. Next question. Okay, ready? Next question. Okay. Chemistry 55, 45 question. Oh, yeah, good question. Good area, guys. Given diagram represent the component of a transcription unit. Select the correct answer regarding it. Okay. Okay. A, C, D, B. A, B, C, D. Yeah. Come on. Yes. Okay. Come on. A, B, C, D. Come on. Who is A? Who is B? Who is C? And who is D? Who is A? Who is B? Who is C? Who is D? A is. Come on. A is promoter. A is promoter. Two options are given. A is promoter. B is terminator. Yes. B is terminator. But next question is how to select a template and how to select a coding strand. Okay. Template and coding strand. Template. Who is template? Template is a parental strand. Okay. Template is a parental strand. Okay. Template is parental strand. The polarity of the template. Actually, you know, the RNA synthesized from 5' to 3' direction. Okay. RNA synthesized from 5' to 3' direction. Who is parent? Parent is just opposite. 3' to 5' is a parental strand. Okay, 3' to 5' is parental strand or template strand. 3' 5' strand is template. The RNA synthesized from 5' to 3' direction. Who is template? 3' 5' is a template. Come on, 3' 5' is a template strand and 5' 3' is coding strand. Which is answer? 3 to 5'. Come on, option C is template strand and D is coding strand. Option C is the answer. Good question, guys. Good question. Super question. Okay, C. Yeah, in Fatima Selva. Yeah, C is the answer. Okay, Angel. Okay, good. Okay, good question, guys. 45 is good question. Okay, next question. The 46, question number 46. Transcription unit. Yeah, next question. Transcription units. What is transcription unit? Come on. 46, transcription units. Start with the data box. Oh, data box mean thymine, adenine box. No. Starts with a palindrome region and end with the rho factor. No. Start with the promoter region and end with the terminate region is called a transcriptional unit. Is a definition of transcription unit. The transcription unit contain promoter and terminator with structural gene is called transcription unit. Okay. Promoter, terminator, between promoter and terminator contain structural gene. Okay, it's called a transcription unit. Option C is the answer. Okay, ready? Option C is the answer. Good. Ready? The next question, the 47. Question number 47. Okay, during transcription, the site of DNA molecule at which RNA polymerase bind is called. During transcription, the site of DNA molecule at which RNA polymerase is bind. Yeah, it's a simple question. Yeah, where is RNA polymerase bind? During the transcription, the DNA molecules at the site of DNA molecule which RNA polymerase bind at called promoter, regulator, receptor, enhancer. Which is answer? Yeah, which location? It's bind? Which bind? During the transcription, the site of DNA molecule at which RNA polymerizer bind is called promoter. Okay, the promoter during the transcription, the RNA polymerase bind at promoter. Okay, bind at promoter, transcription initiate. Which option A is answer. Okay. No, 47 receptor. No, guys, not a receptor. It's a promoter. Okay, it's a promoter. Yeah, not a receptor. Not a receptor. Is a topic is not here. It's a promoter. Promoter is bind. Okay, promoter. During the transcription, the site of DNA molecule at which RNA polymerase is bind is a promoter. I just show that section. Not confusing. Okay, given here. Okay. Look here, guys. Okay, during the transcription is the initiation of transcription. Okay, initiation of transcription. It is the promoter. The RNA polymerase is bind at the promoter. Okay, guys. The RNA polymerase is bind at the promoter. Okay, the RNA polymerase bind at the promoter. The process is called initiation. The transcription is initiated because option A is answer. It's a promoter. It's not a receptor. Okay. Okay, promoter. RNA polymerase is bind at the promoter. Okay, ready? Okay, promoter is answer. The 48. Next 48. Question number 48. Come on. The polycystonic messenger RNA, because mRNA usually occurs in bacteria? No. Prokaryotes? No. Because actually in the prokaryotes only form polycystone. Oh no, guys. Oh, I'm sorry. The polycystonic mRNA. You know what's the meaning of polycystonic mRNA? A single mRNA contain more gene information. Okay, the more gene information. Okay, a single mRNA contain more gene information. Okay, I give you just examples. Okay, guys. Okay, it's a very important question. It's a very difficult area. Okay, guys, look here. It's a lac operon. Example is lac operon. Example is lac operon. Yeah, okay, guys. Look here. It is a lac mRNA. Lac mRNA means it is a polycystonic mRNA because a single mRNA contain. Students, please look here. It's a very important question. Okay, it's the most difficult question. Okay, guys, look here, guys. It's a lac mRNA, which means it is polycystonic mRNA. This single mRNA contain the more gene, Z gene, Y gene, A gene. Okay, the 3 gene information contain a single mRNA is called polycystonic mRNA. It's called a lac mRNA. The lac mRNA contain 3 gene information, Z gene information, Y gene information, A gene information, because the 3 genes are expressed: beta galactosidase, permease, and transacetylase. Okay, the 3 enzymes are produced because the 3 gene is activated through a single mRNA. It is polycystonic mRNA present in bacteria, and prokaryotes. Okay, the polycystonic messenger The polycistronic, cistron means gene, okay, cistron means genes, the polycistron, the more gene, messenger RNA, mRNA, usually occurs in bacteria and prokaryotes, both A and B is answer. Good, okay, ready? 48 is D is answer. 48 is D, okay, because prokaryotes is bacteria and prokaryotes, okay, not eukaryotes. Eukaryotes contain only monocistronic, the monocistronic mRNA, okay. It's a very important question, very important your question, guys, okay, ready? Okay, eukaryotes only monocistronic mRNA, because a single mRNA contain only one gene information, only a single gene information, okay, in eukaryotes. It's a very difficult question, guys, okay, ready? I thought you understood. Angel and sir, the poly physics chemistry MCQ discuss chemo. Actually, Fatima Selva. Actually, നീ മലയാളിയാണല്ലോ, ഞാൻ പറഞ്ഞുതരാം. ഞാൻ ഉദ്ദേശം എന്ന് വെച്ച് കഴിഞ്ഞാൽ ഇതെൻ്റെ വീട്ടിൽ, ഇറ്റ്സ് മൈ ഹോം, ഞാൻ റൂമിൽ സ്റ്റുഡിയോ സെറ്റ് ചെയ്ത് ഞാൻ എൻ്റെ എല്ലാ ക്ലാസ്സുകളും ഒഴിവാക്കി എന്ത് ചെയ്യാണ്, ഞാൻ ഒരു എടുക്കുകയാണ്. ചുരുക്കി പറഞ്ഞാൽ വേറെ ഫാക്കൽറ്റീസിനെ കൊണ്ടുവന്ന് ക്ലാസ് എടുക്കാൻ ഞാൻ ആലോചിക്കുന്നുണ്ട്. ഐ തിങ്ക് നെക്സ്റ്റ് മന്ത് കേട്ടോ, ജനുവരി ഓർ ഫെബ്രുവരി, ബിഫോർ യുവർ നീറ്റ് എക്സാം. ഓക്കെ, ഐ ജസ്റ്റ് ട്രൈ ദ ഓൾ ക്വസ്റ്റ്യൻ പേപ്പർ, ഫിസിക്സ് ആൻഡ് കെമിസ്ട്രി. ഓക്കെ, പിന്നെ ഞാൻ ഉദ്ദേശിച്ചത് എന്താ അറിയോ സെൽവ, ഞാൻ ഉദ്ദേശിച്ചത് 5 മെറ്റീരിയൽസ് ഉണ്ട്, 5 മെറ്റീരിയൽസ്. ഞാൻ ഇടയ്ക്കിടയ്ക്ക് എല്ലാ വീഡിയോസിലും കാണിക്കാറുണ്ട് 5 മെറ്റീരിയൽസ്. NCERT ഫിംഗർ ടിപ്സ്, NCERT പഞ്ച്, മാസ്റ്റർ NCERT, അതുപോലെ A to Z ഈ സാധനമൊക്കെ. A to Z ഈസ് എ ഗുഡ് മെറ്റീരിയൽ. A to Z സിങ്കേജിൻ്റെ A to Z, നീറ്റ് ഗൈഡ് കണ്ടോ, നീറ്റ് ഗൈഡ് റെഡി. അപ്പോൾ 5 മെറ്റീരിയൽസിലെ ക്വസ്റ്റ്യൻസ് ചെയ്യാനുണ്ട്, അഥവാ ഒരു ചാപ്റ്ററിൽ നിന്ന് 500 ക്വസ്റ്റ്യൻസ്. അപ്പോൾ 500 ക്വസ്റ്റ്യൻസ് ചെയ്യണം ഒരു ചാപ്റ്ററിൽ നിന്ന്. ആൻഡ് ദെൻ NCERT ലൈൻ വിത്ത് ലൈൻ ആയിട്ട് എന്ത് ചെയ്യണം, വായിക്കണം. അപ്പോൾ ഇതിന് സമയം, അഥവാ ഇനി നീറ്റ് എക്സാമിന് അല്ലേ, ഓൺലി 132 ഡേ, ഓക്കെ, 132 ഡേ ആണ് ഞാൻ നീറ്റ് എക്സാമിനുള്ളത്, വെറും 132 ഡേ. ആ ദിവസത്തിനുള്ളിൽ ഇത്രയും കാര്യങ്ങൾ ചെയ്ത് തീർക്കാൻ പറ്റുമോ എനിക്കറിയില്ല. അപ്പോൾ അതുകൊണ്ടാണ് എന്ത് ചെയ്യുന്നത്, ഞാൻ ഇത് മാത്രം ഫോക്കസ് ചെയ്യുന്നത് കേട്ടോ. ഫാത്തിമ സിൽവ ബയോളജി മാത്രം ഫോക്കസ് ചെയ്യും, ബിക്കോസ് അത്രത്തോളം എടുക്കാനുണ്ട്. ഓക്കെ, റെഡി? ഓക്കെ ഗായ്സ്, അപ്പോൾ ബാക്കിയിലേക്ക് പോകല്ലേ, റെഡി? ഫാത്തിമ സെൽവക്ക് ഞാൻ പറഞ്ഞ് മനസ്സിലായിട്ടുണ്ടാവും. അപ്പോൾ ചുരുക്കി പറഞ്ഞാൽ ബയോളജിന് ഇഷ്ടം പോലെ എടുക്കാനുണ്ട്. പിന്നെ ഞാൻ ഇങ്ങനെ ഫിസിക്സ് എടുക്കുന്നതാണ് ആലോചിക്കുന്നത്, പക്ഷേ എന്ത് ചെയ്യാം. ഓക്കെ, ഐ എം ട്രൈ ദാറ്റ്. ഓക്കെ, റെഡി? ദ ഗിവൺ ഫിഗർ റെപ്രസെന്റ് ദ പ്രോസസ് ഓഫ് ട്രാൻസ്ക്രിപ്ഷൻ ഇൻ ബാക്ടീരിയ. ഓക്കെ, റെഡി? Okay. The given figure represent the process of transcription in bacteria. The given figure represent the process of transcription in bacteria. Okay, good question. Oh, good. Yes. Who is A, B, C? Yeah. Only 1, 3, 2. Yes. Okay, ready? Just a calculation. Today December 22. Okay, look here guys. Today December 22. How many days is pending here? Only 9 days. Okay, 9 days pending. 9 days actually. 9 days. January 31, February 28, and March 31. March April is 30, and May 3 is your exam. May 3 is exam. Just calculate. 31 is 9, 40, 58, 132. I thought you understood. Hello? Okay. Thank you. Okay, understood. Ready? Okay, only 132 day is pending your NEET exam. Okay, അപ്പോൾ നീറ്റിന് അത്രയും ദിവസമേ ഉള്ളൂ. അപ്പോൾ എന്ത് ചെയ്യുക, you studies very comfortably and your studies very smartly. Okay, ready? Next question, come on. Which is answer? A is DNA, No, no A is DNA, not A is DNA. A is RNA. Okay, A is RNA. Two options are given, B and C. Yeah, A is RNA, and B is, who is B? B is RNA polymerase. Yeah, B is RNA polymerase. B is RNA polymerase. Two options are given, B or C is answer. Okay, and who is B is RNA polymerase? Who is C? Who is C? C. First of all, you understood the picture. Yeah. First of all, you go to your anxiety test book and you, first of all, you, the picture is very, very, very important. Yes. Who is this? Oh, no. Yeah. Okay, it is a figure. It is RNA and RNA polymerase and it is rho factor. Okay. It's a sigma factor. It is a sigma factor. It's a sigma factor. It's a sigma factor. Promoter. The same figure. It's a promoter, RNA polymerase, and it is sigma factor. And it is RNA polymerase, and it is rho factor. Okay. Yeah. Which option? Yeah. RNA polymerase, and it is rho factor. Option B is the answer. Okay. Option B is the answer. RNA polymerase and rho factor. Option B is the answer. It's a simple question. Okay, simple question. Option B is the answer. Okay. The next 50 question. Refer the given diagram. What do it represent? Yes. Given diagram, what does it represent? Transcription in prokaryotes. No. Actually, what it is represented? Actually, it is, what it is? It is, yeah. It is hnRNA. It is hnRNA. Okay. Heterogeneous nuclear RNA. Okay. It is heterogeneous nuclear RNA because this RNA contains both exons and introns. Exon is coding sequence. The intron is non-coding sequence. Okay. After the splicing, the mRNA is produced. Okay. The hnRNA converted to mRNA. Actually, this process is called splicing or modifications. Okay. Come on. What this picture is represented? Transcription in prokaryotes? No. Transcription in eukaryotes? No. Translation in prokaryotes? No. Translation in eukaryotes? No. No. Which is the answer? Transcription in prokaryotes? No. Transcription in eukaryotes? Yeah. Actually, it is the part of transcription. This option is correct. It is a part of transcription. Actually, this takes place in eukaryotes, not a prokaryote, because in the case of prokaryotes, after transcription directly produced. Okay guys, in prokaryotes, after transcription directly produced mRNA from DNA. But in the case of eukaryotes, after the transcription, produced DNA to hnRNA, and then once again guys, okay, in eukaryotes the hnRNA produced from DNA, okay, in eukaryotes. But in the case of prokaryotes, mRNA directly produced from DNA. But in the case of eukaryotes, hnRNA produced from mDNA, and after hnRNA converted into mRNA. Actually, this process takes place in eukaryotes. Is a transcription. Okay. Which options are answer? Option B is the answer. Ready? It's a simple question. The next question. Okay, ready? Come on. In transcription of eukaryotes, the heterogeneous nuclear RNA, because hnRNA is transcribed by RNA polymerase 1, no. RNA polymerase 2, yes. And RNA polymerase 3, no. And D, all of this, no. Option B is the answer. Okay. You take your NCERT test book. I show you the answer. Yeah. You take your NCERT book. Look here guys, is the answer. It's a very, very important section. The very, very important section. Yes, this answer. It's this area. It's a very important. And the RNA polymerase 1. Okay, the RNA polymerase 1 is RNA polymerase 1 polymerase the rRNA. Which rRNA? 28As. Okay, 28As, 18As, and 5.8As. Were the RNA polymerase 3 is polymerized tRNA, 5 srRNA and snRNA, small nuclear RNA, and RNA polymerase 2. Okay, the RNA polymerase 2 transcripts is synthesized mRNA, that heterogeneous nuclear RNA. Okay, the hnRNA synthesize RNA polymerase 2. Okay, the RNA polymerase 1 is synthesized 28S, 80S and 5.8 rRNA, and RNA polymerase 2 synthesize tRNA, 5 srRNA and snRNA. It's a very important your section. Okay, very important. And first of all, you study this area, your NCERT book. Complete your study, okay? And go into your question. Okay, let's go into your questions. Come on. Oh my goodness, guys. I'm sorry. Where is this? Oh, I'm sorry, guys. I'm sorry. It's given. I think question number 52. Last discuss question number 52. Okay. Yeah, 51B. Okay, 52. Come on. So now loading. Just wait. Coming. It's a big PDF because guys, come on, just wait. Come. Just a small, take 1 minute, okay? Come on, man. Oh my goodness. Come in, guys. Just wait. Okay. What happened? Nice. Oh, damn. It's the principle of inheritance and variation. It's a molecular, yes. Okay, ready. Let us start the question number 50. Yeah, 53, 52. 50. Oh, no. Yeah, 50. Yeah. In translation, eukaryote heterogeneous nuclear RNA HNR translated by, yes, RNA polymerase 2. Yeah, I thought it is a last discussed question. RNA polymerase 2 is answer. Okay. HNRNA synthesized RNA polymerase 2. Okay. Okay, ready. And the next question. Next question. The methyl guanosine triphosphate is add at 5' end. Simple question, guys. Oh. Just wait. The methyl guanosine triphosphate add at 5' end is called. Yeah, at 5' end is called. Methyl guanosine triphosphate add you add to the 5' end of HNRNA process is called. So simple, capping, splicing, tailing. Yeah, which is, come on, capping. The methyl guanosine triphosphate is added to the 5' end of HNRNA is process of, it's called, yeah, simple, it's called capping. Okay, it's called capping. Okay, the methyl guanosine triphosphate added to the 5' end is called capping. The three process, capping, splicing and tailing. 52, none of this, no, it's a capping. Okay, capping is the process of, what is process? The methyl guanosine triphosphate is added to 5' end HNRNA process is called capping. Okay, the capping is answer. Okay, you take your NCERT test, so I show that, yes. Okay, it is capping. It is capping. Capping means the three methyl guanosine triphosphate added at 5' end of HNRNA is called capping. I thought you ready? Okay, ready? Option, yeah, option B is answer. Okay. First of all, you read NCERT with a line. Okay, capping is answer. The next question. Select the correct statements and regarding the process of transcription in eukaryotes. Okay, transcription in eukaryotes. 53. Question number 53. Transcription in eukaryotes. Come on. 53. Okay. Which statements are correct? Correct statement. Yes. Okay. Correct statements. The strand of DNA, the strand of double-stranded DNA, which take part in transcription process is called coding strand. No, is a template strand. Okay. The strand of DNA, double-stranded DNA, take part in transcription process is called not a coding strand, it's a template strand. Okay, it's incorrect. The enzyme RNA polymerase can catalyze polymerization only one direction, 5' to 3' is correct. The unusual nucleotide methyl guanosine triphosphate is added at the 5' end of HNRNA during capping. Yes, is correct. During the tailing process, adenylate residues, because 200 to 300 adenine add at the 3' end of template independent. Add at 3' end at a template independent. The statements are correct. Yes, the statements are correct. Okay. First of all, you read NCERT test with a line. Okay, you can see the three statements your NCERT. The three statements are correct. Okay, 2, 3, 4, 2, 3, 4, 2, 3, 4 is a 2, 3, 4 option. C is the answer. It's a good question, guys. Good question. Okay, option 3 is the answer. Okay, ready? I thought you understood. It's a simple question. Okay, ready? Go next question. 53. The option C is the answer. Okay, very good. Angel, yeah, Angel and Fatima Selva. Okay, good, ready. And then next 54. That's the 54 question. 54. Numbered question cheyato. Yeah, maximum question cheya. Nallu neat kittulla. Allade NCERT kutharnu vaaichondu kaarilla. Maximum question. You solve maximum questions. The maximum questions. Kado. The maximum question. The itrayum questions nammal cheyanam. 5 materials. NEET guide 1. Kado. It's a NEET guide. And A to Z is a very difficult question, super question. A to Z is a Singage question, it's a very, very difficult question. And Master of NCERT good questions, and Punch NCERT Punch is a good question, maximum question. Okay, I thought biology is a very easy subject, but question is a very difficult. Okay, total 90 questions, 45 zoology, 45 botany, total 90 questions. With crack the 90 questions within 1 hour, 60 minutes, 60 minutes, 90 questions. This is our mudravakyam. This is our challenge. This challenge is NEET simple. How to NEET crack very easily? NEET easily crack the answer is the biology question, the 90 question crack within 60 minutes. 60 minutes NEET crack. The maximum questions are solved. Questions maximum solve. Maximum questions solve. Ready? In eukaryotes, look. In eukaryotes, the processing of primary transcript involve. In eukaryotes, the processing of primary transcript involve removal of intron, capping 5' and tailing polyadenylate at the 3' and all of this. Yeah. In eukaryote, processing primary transcript. Who is primary transcript? That is hnRNA. The hnRNA. Okay. Okay. In eukaryotes, processing of primary transcript. Who is hnRNA? Involve removal of introns. Yes. Capping of at 5' end. Yeah, it's a capping. The removal of introns is called splicing, and the tailing, the polyadenylate at 5' end is called tailing. Okay, capping, splicing, and tailing. Okay, it's option D. All of those. Ready? Which is answer? Option D is answer. Okay, ready? Then ready. Next question. The next question. Okay, next question. The fully processed hnRNA is called. The fully processed hnRNA called. The fully processed hnRNA is called mRNA. Yes, the fully processed hnRNA is called. Yes, called mRNA. Yeah, is called mRNA. Okay, option A and B is mRNA. mRNA is transported out of the nucleus. It's a transporter out of the nucleus into cytoplasm for translation. Option A is answer. It's a correct statement. Okay, ready? So good question guys. Good question. Okay, the fully processed hnRNA converted into mRNA is transported out of the nucleus into cytoplasm for translation. The protein synthesis take place in the cytoplasm. The protein synthesis take place in the cytoplasm. The ribosome is present in the cytoplasm. Okay, the hnRNA convert into mRNA present in the nucleus. hnRNA convert into mRNA, the process is called splicing. The splicing is take place in the nucleus. After the splicing, the mRNA convert into cytoplasm from nucleus. Ready? The statement is correct. Okay, option A is correct answer. The next question. Identify A, B, C, D in the given diagram of mRNA. Who is A, B, C, D? Ready? Okay, who is A, B, C, D? Okay, ready? A, B, C, D. Okay, A, B, C, D. Who is A? A is methylguanosine triphosphate. Methylated cap. Three options are given. B is removed. Okay. Option B. Who is B? Who is B? You know? You take your NCT test book. Okay, come on. Who is B? 55, yeah. 55, okay. Okay. Who is B? Exons or intron? It's the introns. B is introns. Initiated codon? No. And non-coding region is, it is not coded region. B is introns. B is introns. Who is intron? Intron is non-coding region. It's a not coded protein. And C is exons. C is exons. Exons code for protein. Okay. Exon is code for protein and introns non-code for protein. Okay. Okay. It's a removed part. It's a waste. It's a waste material. B is a waste material. Okay. It's a non-coded region. Okay. Okay. It is non-coded region. And who is C? C is exon coding region. C is exon coding region. Who is D? D is poly a tail. It's a tail. Come on, which is option? Option C is the answer. It's a methyl cap and it is non-coding sequence because B is introns and C is exons coding sequence and is a tail. Option C is the answer. So simple question. Okay, very simple question. First of all, you read NCERT mustly. NCERT book. NCERT is mustly you read. You can crack questions. The line of NCERT. Okay, ready? Okay, ready? Option C is the answer. Then that's 57. Okay, 57. Refer to the given sequence of the steps and select the correct option. Given sequence and step, the select correct sequence. Okay. DNA convert into hnRNA, hnRNA convert into mRNA, mRNA convert into protein. Come on. DNA convert into hnRNA. Process is called actually it's a process taking place in eukaryotes. So okay, only eukaryotes, not a prokaryote, because prokaryote hnRNA is absent and eukaryotes hnRNA is present. Okay, come on. Okay, DNA convert into hnRNA. DNA convert into hnRNA process is called a replication. Not a replication. No replication. It's a transcription. Yes, two options are given. Transcription. C or D is answer. Okay. The HNR converted in mRNA. Replication splicing. It's a splicing. His answer is given. It's a simple answer. Answer is given here. It's very simple. Okay, first of all, it's a transcription, not a replication. Okay. Okay, two options are given, C or D. Next, HNR converted in mRNA. HNR is a splicing. Yes, splicing. Okay, option not option D, option C is answer. And let's say mRNA protein is a translation. Option C is answer. Okay, ready? Very simple, very simple question. 57C is answer. Okay, Angel. Okay, Angel. Okay, ready? Come on. Selva. Okay, Selva. Ready. Good. Come on. Okay. I thought you ready for 100 question. Complete it. 100 question complete it. 100 ready le. Talariddo. Talarndu kaariya. Nammal talarchi innu nikkula. Okay, not tired. Come on, let's go. 100 question. Okay, come on. Okay. Oh, next question, genetic code. Wow, most important section. Yeah, most important section is a genetic code. Or Rakshila. Genetic code. Come on. It's a simple question. Yeah. Simple question. Come on. It's a genetic code. Genetic code is the most valuable question for molecular basis of inheritance. The most important question, the molecular basis of inheritance. The mustily and surely it's a very important question for molecular basis of inheritance because genetic code. Come on. Which codon are respectively code for proline and valine in amino acid? Yeah, it's the most difficult question. Yeah, it's a difficult because a NEET students, a NEET aspirant students, please you study the complete table of genetic code, which is very stable. Come on, you completely study this table because, come on guys, to completely, okay, otherwise you not crack NEET exam. Otherwise, you cannot crack NEET exam. നിങ്ങൾ ഭയങ്കര ഇമ്പോർട്ടൻ്റ് ആയിട്ട് പഠിക്കുന്ന സാധനം. ഇത് പഠിക്കാതെ നീറ്റ് ക്രാക്ക് ചെയ്യാൻ കഴിയൂല. Sure. You want to crack NEET, you first study the complete. Okay, ready? Okay. Without this table, you cannot crack NEET. Otherwise, you pray your God. My God, please. ഇത് തരല്ലേ എന്ന് നമ്മൾ പ്രാർത്ഥിക്കുക. Otherwise, no രക്ഷ. Okay, because they're very important. And you first of all you study the complete genetic code and their code amino acids. Okay, ready? And the first of all, I'm going to question like a okay, which is answer? Come on, which is answer? Which codon respectively code for proline and valine? Yeah, the proline is code word CCU code by proline. Okay, the proline code by CCU and the valine is code by GUU. The option A is the answer. Option A is the answer. Very important section. Okay, students, the first of all option A is the answer. Okay, the first of all you must still study the table. No excuse. So very important. Okay, ready? Next question. Next step. The 59. The amino acids which are specialized by only single codons are. Okay, the next question. Amino acids. Next question. Amino acid which are specified by only single codon. Only single codon. Example, come on, which is answer? Only single codon. Two amino acid coded only a single codons. Who is this? Okay, I think you are difficult. You go your answer. You select. Or one amino acid coded only. Come on, which is answer, guys? Okay, you can crack. Come on. One amino acid. Example, look here, guys. It's a methionine. Methionine coded only AUG. Is there another one amino acid that coded only? One codons. Come on. Not a phenylalanine, leucine. Okay. Valine, alanine, threonine, proline, serine. Okay, histidine, glutamine, no. Arginine, serine, cysteine. TRP? Is it TRP's answer? No. TRP is, where is another? TRP. TRP is another. Where is, no, only one TRP. His answer is yes. Methionine and tryptophan. Come on, which is answer? Yeah, given the answer. Which is the answer? His answer is methionine and tryptophan. Option 59. Answer. Yeah, 50. Okay, option B is answer. It's a simple question. Ready? So this is a neat question. Okay. Okay, ready guys. Tryptophan and methionine is answer. Only one single codons. Amino acids which are specified only single codons. The tryptophan and methionine is answer. By heart. You study the by heart. Ready? Okay, ready. The next question. Read the following statements. One codon coded. Okay. Read the following statements. Okay. Okay, guys. Look here, guys. One codon coded only one amino acids. One codons for only one amino acid. Okay. Come on. Which is answer? One codon coded for only one amino acid. Which is answer? What is called? Degeneracy, universal or unambiguous? One codon coded for only one amino acid. The statement is called degeneracy. The statement is called degeneracy. The statement is called degeneracy. The option 1. Two options are degeneracy. No, not degeneracy. It's universal. No guys, something problem. Give the suitability for the character for the genetic code as per the above statements. Okay. Degeneracy. What is degeneracy? Some amino acid. Look guys. One codon coded for only one amino acid is called unambiguous. That statement is correct. Yes, unambiguous. It's unambiguous. It's unambiguous. So it's unambiguous. It's unambiguous. It's only one. Two options are unambiguous. Ready? Okay, option B or C is answer. It's a very difficult question, guys. B or C is answer. Okay, one codon coded for only one amino acid is called unambiguous. Okay. Unambiguous. What is unambiguous? One codon coded for only one amino acid is called unambiguous. Two options are given. Unambiguous is 1, 1, 1. Okay, ready? 1, 1, 1. Unambiguous. Some amino acid coded by more than one codon. Some amino acid coded by more than one codon is called. Some amino acid coded more than one codon is called. Is a universal. Is called a universal. Universal? No, not universal. Oh, man, it's a very difficult question. Come on, it's a universal. Yeah, it's a universal. Some amino acids are coded by more than one codon is called universal. Okay, ready? Correct. It's a universal. And next, from the bacteria to human, you would code for phenylalanine. Okay, it's called. Guys, I'm very. It's very confusing question. Come on, you give your answer. Everybody, come on. Angel, come on. Which is answer? You can solve. Here, first of all, one codon coded for only one amino acid. Yes, the statement is unambiguous. Okay, unambiguous. Two options are given. Okay, the next. Some amino acid coded by more than one codon is called degeneracy. Yes, the statement is called degeneracy. Option 2. Okay, degeneracy. Where is option 2? 2. Yes, 2. Degeneracy is given here. It's a degeneracy. And option 3. From the bacteria to human, you would code for phenylalanine. The statement is called universal. It's a universal. The option C is the answer. Okay, ready? Option C is the answer. So very difficult question. You first of all, you read. It's a confusing statement. You first of all, you study the three statement is very important. Okay, ready? നന്നായിട്ട് പഠിക്കുക ബിക്കോസ് നന്നായിട്ട് അങ്ങോട്ടും ഇങ്ങോട്ടും തല ഇരുന്ന് എന്ത് ചെയ്യും മാറും ബിക്കോസ് 2 മിനിറ്റ് 3 മിനിറ്റ് എടുക്കും 1 or 2 or 3 or minute solve this question. Okay, and first of all you study by heart these statements. Ready അല്ലേ? 60. The answer option C. ഒന്നുകൂടി പറഞ്ഞേ. Okay, what is degeneracy? Degeneracy is some amino acid are coded by more than one codon. What is degeneracy? Some amino acid are coded by more than one codon. Okay, what is universal? From the bacteria to human, you would code for phenylalanine. Okay, in the case of human, in the case of bacteria, you code phenylalanine because it is universal. The genetic code is universal. What is unambiguous? What is unambiguous? The one codon coded for only one amino acid, that is unambiguous. Okay, you study the three statements, are very important, otherwise very confusing. Ready? The next question, 61, question number 61. Some amino acid are coded by more than one codon, hence the genetic code is called, or already discussed. Which is answer? Some amino acid are coded, more than one codon is called degenerate. Ready? It's called degenerate. Your NCERT, pure NCERT question. Ready? It's called degenerate. Some amino acid are coded by more than one codon is called degenerate. Option B is the answer. Ready? Okay, next question. Which out of the following statement is incorrect? Okay, which statements are, come on guys, come on, join with me, come on. Next question. Which out of the following statement is incorrect? Which statements are incorrect? Come on, next question. Which statement are incorrect? Genetic code is, which out of the following statement are incorrect? Okay, genetic code is ambiguous, genetic code is degenerate, correct, genetic code is universal, correct, genetic code is non-overlapping, is correct, ready? Following statement is ambiguous, it's unambiguous, it's not ambiguous. It's a statement is incorrect statement, genetic code, because it's a ambiguous, no, it's unambiguous. Okay, it's unambiguous, not ambiguous. Okay, understood? It's a very important. It's a very difficult question, guys. Not ambiguous, it's unambiguous. Okay, just confusing. Okay, ready? Genetic code is ambiguous. Statements are incorrect. Oh my goodness. Okay, ready? It's a very difficult question. Okay, understand? It's a very difficult question. Okay, the statement is option A is incorrect statement. Ready? The three codons which result okay, next question. Come on. The three codons which result in the termination of polypeptide chain synthesis are, because who is stop codon? That's the answer. Who is stop codon? The stop codons are three: UGA, UAG, UAAA. Okay, UGA, UAG, UAAA. It is a stop codon. Okay, who is stop codon? UGA. Come on. Fatima Selva, come on. UGA, UAG, UAAA are the stop codons. UGA, UAG, UAAA. Come on, who is stop codon? UGA, UAAA, UAG. No, it's a no. UAA, UAG, UAG, UGA, UAG, UAAA. Option B. B is answer. Okay. UAAA, UGA. No. No. Okay, which is answer? Option B is answer. Stop codons are three: UGA, UAG, UAAA are the stop codons. Ready? Okay. Write the sequence of nucleotide in the given segment of mRNA and the represent amino acid sequence in the polypeptide chain. Okay. Read the sequence of nucleotides in the given segment of mRNA and the respective amino acid sequence in the polypeptide chain. mRNA is given, the polypeptide is given. Okay, the nucleotide sequence of the DNA template strand from the which of this mRNA was transcribed by. Oh, it's a good question, guys. It's a good question. Come on, this guys, look here, guys. AUG is methionine. Okay, it is mRNA. Is mRNA. Okay, mRNA, the opposite, which the mRNA was transcribed is. Okay, you write therefore. First of all, you write DNA. Okay, 64 question. Come on, which is answer? Which is answer, guys? Simple question. Yeah, simple. It is AUG, the opposite step. It is mRNA, the opposite is template strand, DNA. A opposite T, U opposite A, G opposite C, U opposite U is a thymine, opposite adenine. A opposite is a, option is here. This option is here. Yes, it's a TAC and okay, TAC, A. It's a but no, option A is answer because it's a DNA strand. Okay, the DNA is a TACAA, but RNA is, mRNA is AUGUU. Okay, it is polypeptide. You write your DNA. The polypeptide is given, mRNA is given. You write DNA. It is DNA. Okay, option A is answer. Simple question. Okay, option A is answer. Fatima Selva, are you okay? Ready? Ready? Come on, next question. Okay, ready? Nirthano, Madeo. Oh, 11 mani aile. Tamone. Question number 65. Selva, നമുക്ക് നിർത്തിയാലോ? ബാക്കി നമുക്ക് ഇനി ഈവനിങ് എടുത്താലോ? 11 മണിയായി. Oh, 64. I'm very tired. 64. 64 question. 64, 65 and ബാക്കി ഇനി പിന്നെ ചെയ്യേണ്ടി വരും. സെൽവ, are you okay? ബാക്കി ഞാൻ പിന്നെ ചെയ്യട്ടെ. സെൽവ, നീ ഓക്കെ ആണോ? നീ കണ്ടിന്യൂ ചെയ്യാനുള്ള ആഗ്രഹം ഉണ്ടെങ്കിൽ ഞാൻ കണ്ടിന്യൂ ചെയ്യും. എനിക്ക് ഒരു ക്ലാസ്സിന് പോകാൻ ടൈം ആയിട്ടുണ്ട്. ഓക്കെ, ആക്ച്വലി ഐ യു റെഡി? ഏഞ്ചലൊക്കെ വന്നോ? ഏഞ്ചൽ, കമോൺ, ഏഞ്ചൽ വന്നോ? സെൽവ വന്നോ? ഏഞ്ചൽ ആൻഡ് സെൽവ, come on. Ready. I'm waiting for your reply. Okay, guys. Come on. So, offline class പോകുന്നുണ്ടോ ഇപ്പോൾ? യാ, ജസ്റ്റ് ഞാൻ പോകുന്നുണ്ട്, ഇടയ്ക്ക് ക്ലാസ്സിന് പോകുന്നുണ്ട്. യെസ്, പോകുന്നുണ്ട്. എന്താ ചെയ്യണേ? കണ്ടിന്യൂ ചെയ്യണോ? ഓക്കെ, കണ്ടിന്യൂ. 11 o'clock ആയി. 11 മണി. How many questions? 64. ഓക്കെ ഗായ്സ്, കണ്ടിന്യൂ ചെയ്യാം. ഇത് തീർക്കാം. കണ്ടിന്യൂ. ഓക്കെ, ഞാൻ കണ്ടിന്യൂ ചെയ്യാം. എനിക്കൊരു ക്ലാസ്സിന് പോകുന്നുണ്ട്, കുഴപ്പമില്ല. കണ്ടിന്യൂ. ഓക്കെ, കണ്ടിന്യൂ. ഏഞ്ചൽ കണ്ടിന്യൂ ചെയ്യണോ? കണ്ടിന്യൂ ചെയ്യാം, തീർത്തിട്ട് പോവാം. ഓക്കെ, അല്ലെങ്കിൽ അത് ചെയ്യൂല അല്ലേ? മുന്നോട്ട് പോകാം. ഓക്കെ, നെക്സ്റ്റ് ക്വസ്റ്റ്യൻ. Read the sequence of nucleotides in the given segment of mRNA and the respective amino acid sequence in the polypeptide chain. mRNA ഇത് നമ്മൾ ചെയ്തല്ലേ? ഇത് ചെയ്തല്ലേ അല്ലേ? The next question. ട്രാൻസ്ലേഷൻ വരാനായി. ഓക്കെ. The mutations that involve addition, deletion or the substitution of single base pair in a gene is referred to as. Good question. Question number 65. ഏഞ്ചൽ, ഞാൻ ഇടയ്ക്ക് പോകുന്നുണ്ട്. ഇടയ്ക്ക് ഞാൻ പോവും. ഞാൻ ഓഫ്ലൈൻ ക്ലാസ്സിന് ഇടയ്ക്ക് പോകാറുണ്ട്. ഓക്കെ, ഇടയ്ക്ക് ഏഞ്ചലേ, ഇടയ്ക്ക് ഓൺലൈൻ ക്ലാസ്സിന് പോകാറുണ്ട്. പക്ഷേ ഞാൻ പൊതുവേ ഈ ഓൺലൈനിലാണ് ഫോക്കസ് ചെയ്യുന്നുള്ളൂ കേട്ടോ. ഓക്കെ, റെഡി? നെക്സ്റ്റ് ക്വസ്റ്റ്യൻ, കം ഓൺ. 65. Question number 65. The mutation that involve addition, deletion or substitution is a single base pair in gene are referred to as which mutation? Is a point mutation. It's a point mutation. Okay. Option A is the answer. Simple question. Option A is the answer because it is a point mutation. The next question. Okay. The sickle cell anemia result from the single base substitution in a gene that is example of. Okay, sickle cell anemia is a result from single base substitution in a gene that is example of. Come on, frame shift mutation? No, and that is point mutation. So frame shift mutation? Frame shift? No, that's a point mutation. Yes. 65 A is 66. A is answer. Ready? Okay, ready. Okay, guys. Okay, ready. The next question. 67. What is true about tRNA? Ready? Next question. What is true about tRNA? It binds with an amino acid at a 5' end. 5' end? No, that's a 3' end. It is called adapter molecules. Oh, guys, it binds with the amino acid at a 5' end. That statement is correct. Something correct or no? It's a no. 3' end. The amino acid is bind at 3' end. It is also called adapter molecule. Yeah, is adapter molecule is correct. It contains a codon at one end which recognizes. It contains a codon that one end which recognizes the anticodon messenger RNA. No, it contains anticodon loop. The tRNA contains anticodon loop that recognize codon of messenger RNA, not anticodon loop. The statements are incorrect. No, which option? B is the answer. 67 answer option B because tRNA is called an adapter molecule. tRNA carried after molecule. Option B is answer. Okay, ready? You store your energy. We can crack NEET. Next NEET. Come on. Okay. Come on. Next. 68. Question number 68. Come on. 68. Next question. 68. The amino acid acceptor end of tRNA. The amino acid not a 5' end, that is 3' end. Anticodon loop. No. Okay. Option B is answer. Simple question. The amino acid acceptor end of tRNA is called a 3' end. The next 69. The differences between mRNA and tRNA is that. Okay, good question. The mRNA is more elaborate 3-dimensional structure than tRNA due to extensive base pairing. The mRNA more elaborate 3-dimensional structure. Guys, this statement is correct? No. So no chance. Okay. No. No, guys. No. The tRNA is more elaborate 3-dimensional structure due to extensive base pairing. Yes, that's correct. tRNA is usually smaller than mRNA. tRNA is usually smaller than mRNA. That statement is correct? Guys, is correct? No, it's correct. tRNA is usually smaller. Oh yeah, tRNA is smaller because mRNA is big. Okay. tRNA is usually smaller than mRNA. Yes, okay. mRNA bear anticodon, but tRNA has codon. No, guys. mRNA bear codon and tRNA has anticodon and mRNA has a codon. Okay, mRNA contain codon and tRNA is anticodon. 2 and 3. Option, which option? 2 and 3. 2 and 3. 2 and 3. 2 and 3 is correct. Difference between mRNA and tRNA. Option B is the answer. Option B is good question. Okay, option B is answer. Yeah, Angel, good. Ho, Angel, 69B. Oh my goodness. Okay, Angel, correct. The next question, translation. Which RNA carry the amino acid to mRNA, the particular codon corresponding to their anticodon? Simple question. Who is this? It's tRNA. Okay, it is tRNA. Okay, ready? Simple. Yeah, tRNA. Option C is the answer. Okay. Ready? Next question. 71. Refer to the given figure. Grow in polymeric chain. Select the option that identify the X and Y. Okay, collect X and Y. Where is X? X. Yeah, X is given, Y is given. Okay. It is X and it is Y. X and Y. Okay, select the which identify X and Y, DNA sequence for serine P and the anticodon for the same amino acid Q. Okay, question is here. Okay. Oh my goodness. Oh, it's a big question. P and Q. What is P and Q? Guys, where is a P? You know? P and Q. P and Q. Where is P and Q? The DNA sequence coding for serine P. The serine. Okay. Guys, serine is okay. This serine is P. Okay. The serine P. Okay. Serine P. Okay. Serine P and the anticodon for the same amino acid is Q. Okay. Q. Serine is P, okay, serine P and anticodon loop Q. Anticodon loop. First of all, it is tRNA, it is alanine, it is code for alanine and serine. It is a serine. The anticodon loop CCC is anticodon loop. Okay, ready. Serine is P, and CCC is anticodon loop of serine. That is Q. Okay. The anticodon for the same amino acid Q. Okay. Oof. Okay. Ready. Which option? Come on. Which option? What a question. Come on. Which question? Okay. Oh, some very complicated question. P and Q. No. So no option is given. P and Q. Serine. The coding for the serine. Oh, coding for the serine. Serine is coded by. Yes. It is alanine. It is GGG. GG is coding for serine. Guys, just wait. TCA. There is a serine. It is a serine actually. Serine. Another. Yo is another serine. Two serine. This serine is actually indicated. Oh, so this question is very difficult. It's a very difficult question. TCA, UCA. Just wait, guys. TCA and UCA. Just P. Where is P? Where is P? First of all, the selected option which identify polarity X and Y, okay, and DNA sequence coding for serine P and the anticodon for the same amino acid is Q. Okay, P. Two serine is given, guys. Question something problem. The question serine is given. Okay. Is alanine. Is a serine. No, is not correct. Something problem. This question is no problem. I will give you question. Something. This is link the question. Serine. But serine is serine is here. But codon anticodon no already. Link something problem. I will question delete here. So go ivaga. Then answer D one maybe chance. D is maybe chance for answer. But that's not linked. So okay, remove it. Something I don't question. Next question number 72. During the translation, activated amino acid get linked to tRNA. This process commonly called during translation. Okay, during the translation, activated amino acid get linked to tRNA. Activated amino acid process is called amino acylation of tRNA. Charged tRNA. Two options are given. Charged tRNA or amino acylation of tRNA. Yeah, charged tRNA or amino acylation. Oh, an answer. Both A and C is answer. Okay. First of all, look here, guys. The amino acid bind with ATP. A amino acid bind with ATP is called activated amino acid. Okay. The activated amino acids bind with tRNA. The process is called amino acylation of tRNA or charged tRNA. Option. Two options are given. D is answer. Okay, guys. Okay, ready. Okay. 72 option D is answer. Next question. Given below are the steps of protein synthesis. Arrange them correct sequence and the select correct option. Okay. Arrange correct options are one. Okay. Come on. Next question. Come on. Which is answer? Come on. Okay, guys. Look here. First of all, codon anticodon reaction between mRNA and amino acyl tRNA complex. Okay. Attachment of mRNA and smaller subunit of ribosome. Okay. Charging and amino acylation of tRNA. Okay. Attachment of larger subunit of ribosome to the mRNA and tRNA methionine complex. Okay. Okay, ready. And linking of adjacent amino acids. Okay. And formation of polypeptide chain. Okay. Ready. And the first identify the first steps. First steps. Which is first step? Charging and amino acylation of tRNA. It is the first steps. Okay. The first step of protein synthesis, charging. Is correct question? Oh, Angel is the correct question. Come on. How to, which is the answer? The growing polypeptide chain, okay. Select the option which identify polarity x and y. Okay, polarity simply marked 5 dash or 3 dash. 5 dash and 3 dash, okay. The DNA sequence coding for serine. DNA sequence coding for serine P, and the anticodon for the same amino acid is Q. Okay. Okay, yes, then. Okay, then okay, and alanine is given. It is alanine. tRNA is anticodon loop is here. Oh my goodness, select the option. It's a serine. Yeah, serine actually is a, it is a serine. Oh my, yes. Oh, this tr, it is alanine is a serine. It's a coded serine. UCC is codon. Yeah, UCC and it's a AGG. Oh, that option is here. UCC and AGG. UCC and UCC and DNA sequence code for serine. Question DNA sequence, okay. It is mRNA. So DNA sequence is uracil opposite. The opposite of uracil is adenine is a guanine. AGG. Okay, AGG. His answer is AGG. AGG? No. So no. How to? Oh, it's not a picture. Oh, NCRT. Okay. Serine. Okay. NCRT. Oh, yes. പക്ഷേ അത് അങ്ങനെ ഒരു ക്വസ്റ്റ്യൻ തന്നു കഴിഞ്ഞാൽ കൺഫ്യൂഷൻ ആവില്ലേ? But okay, serine. Okay, understood. Serine is coded by, two options are given here. Serine. പക്ഷേ ഏഞ്ചൽ അപ്പോൾ പ്രശ്നം എന്താ അറിയോ? സെറിൻ ഒരുപാട് ആൾക്കാർ കുറയുന്നില്ലേ? AGU, AGC is coded by serine, and okay, UCU, UCC, UCA, UCG, AGU, AGC കോഡ് ചെയ്യുന്നത് സെറിൻ ആണോ? ഒരുപാട് സെറിൻസ് വരുന്നുണ്ട്. ശരി പറഞ്ഞാൽ ആണ്, പക്ഷേ സംതിങ് അല്ല, ഒരു തന്നത് സിസ്റ്റായി തന്നില്ല. NCRTയിലെ ടാബിൾ. ഓക്കെ, TCA. പക്ഷെ എനിക്ക് മനസ്സിലായി ഏഞ്ചൽ, നീ പറഞ്ഞ കറക്റ്റാ. പക്ഷെ ഇത്രയും സെറീൻസ് ഇവിടെ വരുന്നുണ്ട്. പക്ഷെ അങ്ങനെ അവർ ഒറ്റയടിക്ക് സ്പെസിഫൈ ചെയ്ത് ചോദിക്കാൻ, ബൈഹാർട്ട് ചെയ്തൊരു കുട്ടിക്ക് പെട്ടെന്ന് കേട്ടോ. ഈ ടാബിള് ബൈഹാർട്ട് ചെയ്ത് കുട്ടിക്ക് ആൻസർ ചെയ്യാൻ പറ്റും. ഇറ്റ്സ് റെഡി. ഓഫ് കോഴ്സ് റെഡി. ഓക്കെ, ചുരുക്കി പറഞ്ഞ ആൻസർ, ദ ആൻസർ ഈസ്, നമുക്ക് ആൻസർ പറയാൻ പറ്റും. ഇറ്റ്സ് ആൻസർ. Okay guys, two options are given, 5 dash 3 dash 5 dash 3 dash. Option D 5 dash 3 dash ആണ്. പിന്നെ DNA സീക്വൻസ്, ദ DNA സീക്വൻസ് TCA. നോട്ട് ഒരിക്കലും ഇങ്ങനെ പറയില്ല, ഇങ്ങനെ വരൂല. USA. സിമ്പിൾ ലോജിക് വഴി നമുക്ക് ആൻസർ കിട്ടും. പക്ഷേ സിമ്പിൾ ലോജിക്, ബിക്കോസ് 5 dash 3 dash 5 dash 3 dash. The DNA sequence is UUU. It's not a DNA sequence. DNA sequence is TCA and UCA. It's a simple logic. Logic answer good. But the questions are ready question. It's a not good question. Something that's something logical problems. Okay, ready? Okay, ready. Next question. Come on. Okay, next question. Yes, ready. But option is only. Oh yeah, I understood. During the translation, but Angel, that's not a logical question, sir. I'm the questions are neat in it. It don't give ready. During the translation, activated amino acid get linked. Both A and C because charge tRNA or amino acylation of tRNA. Option D is the answer. Both A and C. Ready? Next question. Okay, next question. 73. Okay, 73. Given below. Okay, guys. Next question. Next question. Given below. Come on. Next question. Given below. Okay. Given below are the steps of protein synthesis. Arrange them into correct sequence and select the correct option. Okay. The codon anticodon reaction between mRNA. We already discussed this question. Okay, come on. We already discussed this. It is the step of translation. Okay, the first steps of translation is charging or amino acylation of tRNA. It is the first step of. It is the first step of translation. Okay, it is the first step of translation. Give me which is answer. Okay, which is answer? Charging or amino acylation of tRNA. It's the first step of. It's the first step. It's option 3, guys. It is the first step of translation. Charging or amino acylation of tRNA. 3 is first. Only one option of 3. Only one option is 3 because it is D's answer. Confirm. And 2. Attachment of mRNA and the smaller subunit of ribosome. Okay. And then this 1. The codon anticodon reaction between mRNA and amino acyl tRNA complex. And that's 4. Attachment of large subunit of ribosome to the mRNA and tRNA is a methionine complex. Then that's 5. Step 5. Linking of adjacent amino acid. And the 6. The formation of polypeptide chain. Okay, ready? The option D is the answer. Ready? Option D is the answer. Ready? It's a good question. Next question. Come on, guys. A very easy question and very important question for your NEET exam. NEET exam. Okay, guys. Come on. Refer to the given mRNA. Refer to the given mRNA segment which can be translated completely into a polypeptide. Okay, ready? Which of the following codons may correspond with A and B? A and B. Oh my goodness. Correct. First of all, who is A and who is B? A is start codon, B is stop codon. A is start codon and B is stop codon. Who is A? Who is A? Who is start codon? AUG is start codon. AUG is start codon. AUG is start codon. Who is stop codon? Stop codon is UGA. UAG. Come on, UGA. UAG. Come on, UGA. UAG. Option A is answer. Not a GUG. Not a UUU. Option is incorrect. Option A is correct. Refer to the given mRNA segment which can be translated completely into the poly. Okay, A and B is a start codon and is a stop codon. Ready? It's a simple answer. Option A is correct. Okay, ready. Next question. Okay, next 75. Angel correct, you correct, Angel. Next start the 75. UTR are the untranslated region present on untranslated region present on rRNA, no, tRNA, no, and present in mRNA. Okay, UTR are the untranslated region present on mRNA. Okay, ready. Next, any mRNA. Look here, guys. Next. In a mRNA molecule, untranslated region UTR are the present at. That's a good question. It's a good question, guys. In a mRNA molecule, untranslated regions UTR are the present at 5' end before and start codon, 3' end after stop codon, 3' end before stop codon, both A and B. Give me answer. Anybody come on? Come on. It's a simple question. Pure NCERT. Yeah. Fatima Selva, 75C. 75C, okay. Selva, okay. Good, come on. 76. Who is UTR? Who is a UTR? Untranslated region. Come on. 5' and before start codon. Yes, that's correct. 3' and after the stop codon. Yes, correct. Because two options are correct, but answer both A and B is the answer. Okay. Both A and B is the answer. It's a correct option. Both A and B is a correct answer. Okay, 76. Both A and B is the correct answer. I thought you understood. It's a correct, guys. Yes, correct. Ready. The next, which of the following statements is correct regarding ribosomes? Next question. Which of the following? Come on. Which of the following statement is correct regarding ribosome? Which of the following statement is correct regarding ribosome? Okay, ribosome. Come on, ribosome. Okay, ribosome. Most of the cell DNA molecule are stored there. Which of the following statement is correct regarding ribosome? Okay. Most of the cell's DNA molecule are stored there. No. Cell DNA molecule stored in ribosome? No, that statement is incorrect. That's not correct. Complete polypeptide is released from there. Yes. Because the ribosome synthesize polypeptide or protein chain. The complete polypeptide is released from there. Okay. It's a correct answer. It's a correct. The next. 70. Next. The mRNA are produced there. No, guys. mRNA is produced in ribosome? No. It's incorrect. DNA replication take place there. No. Because option B is the answer. Okay, option B, because the complete polypeptide is released from there. The protein chain released from there. Because option B is the answer. Ready? The next, 78. Question number 78. Come on, good question. 78. What would happen if a gene encoding a polypeptide? Good question. Okay, what would happen if gene encoding a polypeptide of 50 amino acids, that 25th codon's UAU is mutated in UAA? Oh my goodness. A super question, guys. So 78 question is a very difficult question. Anybody can give me answer? Anybody? What would happen if the gene encoding a polypeptide of 50 amino acid? Okay, a polypeptide, because a protein chain contains 15 amino acid. The 25th codons UAU is mutated into the 25th codons mutated into okay, is mutated. This codon mutated into what is it is stop codon. It is a stop codons. Okay, it is a stop codon. The 25th codons are stop codon. How many amino acids are formed? Okay, you understood. Otherwise, I draw the figure. If you want, I draw a figure. Okay, just it's a very important question. Very important question. Okay, first of all, look, guys. Okay, it's a 15 amino acids. Okay, a chain contains 15 amino acids. Okay. First of all, a chain contains 15 amino acids. A very important question. Okay, guys. 15 amino acids. Okay, 15 amino acids. Everybody okay? 15 amino acids. But The 25th genetic code is mutated into UAAAA. Who is UAAA? It is a stop codon. The stop codon not coded by amino acids. Okay, not coded by amino acids. Then the 6th, it is 25, and the 26 and UTR, untranslated region. Okay, UTR is untranslated region. Before the stop codons are coded, because it's a 24th. 24th is amino acid is coded. Okay, it's coded, but 25th is not coded and there is not coded. Come on, how many amino acids are formed? It's a 24 amino acids are formed. 24 amino acids are formed because their option says, oh my goodness, guys. Oh. Just wait, guys. 2 minutes, just wait. 2 minutes, just wait. 2 minutes, coming. Just 2 minutes. Question is coming. Just 2 minutes. Okay? Just 2 minutes. 2 minutes. Just 2 minutes. Coming. That question is a very difficult question. The 15th amino acid polypeptide chain. The 25th genetic code is mutated. That mutated UAA, that is a stop codon. Before stop codon, how many amino acids are present? The answer is 24. Okay, the 24 amino acids are present. It's a big PDF because it's a small. Listen. Sure. So just loading. Wait, it's loading. Yeah. It's loading. Coming. Just wait, guys. Just wait. Please wait. Whoa. Coming. Okay, ready? Yeah. 50, question number regulation. Yes, ready. 78 question. Okay, 78 question. Okay. Yes, ready. Come on, give me answer. Which is answer? Which is answer? Yes. Come on. Yes. Yeah. Which is answer? What would happen if the gene, okay, which is answer? A polypeptide. A polypeptide 24 amino acid will be formed. A 2 polypeptide 24, 25th amino acid will be formed. A polypeptide 29 formed. 25 amino acid will be formed. Which is answer? A polypeptide 24 amino acid will be formed because the 25th amino acid converted into, okay, the 25th amino acid is converted. Which option? A. Okay, Angel, super, Angel. ഞാൻ കരുതി ഇത് കുറച്ച് ഡിഫിക്കൽട്ട് ആണെന്ന്. നീ ആൻസർ പറഞ്ഞു സൂപ്പറായിട്ട്. Ready? Okay, ready. 25th option, 27, 80, the option A is answer. The next question, the regulation of gene expression. Okay, next area, next. The regulation of gene expression. The regulation of gene expression occurs at the level of transcription level, yes. Processing level, yes. Translation level, yes. Once again, translation level and transcription, no. Transcription level, processing level, translation level, once again, transporting level, okay, is not here. 4 level, the gene expression in eukaryotes at 4 level, transcription level, transporting level, processing level, translation level. Okay, ready? Option D, all of this. All of this is answer. Ready? Okay, all of this is answer. It is answer. Option D is the answer. Okay, ready? Okay, ready. The next question. Question number 18. During expression, look, Artha. Next. During expression of a operon, RNA polymerase is bind to. During expression of expression of operon, the RNA polymerase is bind to structural gene, regulatory gene, operator and promoter. Yeah, which is option? Yeah, during expression of operon is expressed, the RNA polymerase is bind to promoter. Option D is the answer. Is bind to promoter. Option D is the answer. This is a good question. Okay, during the expression of operon is expressed, RNA polymerase is bind to promoter. Okay, bind at promoter. Okay, I thought you understood. It's a simple question. No, food കഴിക്കണം വിശക്കുന്നുണ്ട് ഞാൻ പിന്നെ അത് നോക്കിയത് കഴിയാതെ only 20 questions. Okay, after I bring foods only 20 questions, so simple questions are coming. Ready. Okay, thank you Angel. Next question. Promoter. Next question. The sequence of structural gene is lac operon. So simple. Then the symbol. The sequence of structural gene in a lac operon is. The sequence of structural gene in lac operon. The sequence of structural gene in lac operon is lac Z gene, Y gene, A gene. Z gene, Y gene. Z gene, Y gene, A gene. Option D is answer. Lac Z, lac Y, lac A is answer. Okay, ready? Is good. The next question. E. coli cells with a mutated Z gene of the lac operon cannot grow in medium containing only lactose as the source of energy because E. coli cells with mutated Z gene, Y gene, A gene. Okay, Z gene is mutated in the lac operon, cannot grow in medium. Yeah, first of all, what's the function of Z gene? Good question, guys. Good question. Which is answer? Come on. First of all, what's the function of Z gene? Z gene produce beta galactosidase. What's the function of beta galactosidase? Z gene, Y gene, A gene. Beta galactosidase convert lactose into gluculata. Lactose. Z gene produce beta galactosidase. The beta galactosidase enzyme convert lactose into glucose plus galactose. Okay, the Z gene is mutated. What happened? The lac operon is constitutively active with the cells. No, they cannot synthesize functional beta galactosidase. Yes, that is answer, because the Z gene is produced beta galactosidase. The beta galactosidase convert lactose into glucose plus galactose. Option B is answer. Option B is the answer. 83. The given figure show the lac operon and is functioning. Select the option which correctly labeled A, B, Z, Y, Z. A, B, Z, Y, Z. Okay, Wagi, come on. Which is answer? It's a good big question. Question number 83. Which is answer? Come on. Which option? Simple, come on. Who is A? A is repressor protein. Okay. B, who is B? Is an inducer. So repressor is an inducer. Okay, two options are A or B is correct. A, B or correct. And Z, beta galactosidase, not a permease. Y is permease. C, Z, transacetylase. Option A is answer. Symbol. So pure NCERT. Okay, 83. Okay, next 84. Which of the following cannot? Next question. 84. Come on, which cannot? Which of the following cannot act as an inducer? Act as an inducer. Glucose act as inducer? No. The galactose act inducer? No. Lactose act as inducer? Yes. Cannot act as inducer. Both A and C. Option D is answer. Glucose and galactose act as inducer. Cannot act as inducer. Option D is answer. It's a simple question. Ready? Ready. Next question. 85. Next 85. Read the given statement regarding the regulation of gene. Okay. Okay. Come on. Read the given statement. Okay. Regarding. Okay. Select it. Lactose act as the suppress for gene expression. Lactose act as the suppressor for gene expression. Act as a no. Galactose act as the inducer for gene expression. Okay. Galactose act as the inducer for gene expression. No. The regulation gene is the one that produces the repressor molecule. Regulator gene is the one that produces the repressor molecule. Yes, correct. A and C are correct. No. Only B is correct. No. Only C is correct. Yes. A and B are correct. No. Option C is answer. Only C is correct. Only C is correct. Ready. Ready. The next human genome project. So simple question. Come on. Fastly. Come on. 86. 86. The human genome consists of approximately 9 into 10 raised to 9 base pair, 62 3 into 9, not a 6. No. Yes, option A is answer. Option A is answer. 86. Option A is answer. 3 into 10 to 9 base pair. Ready? Ready. Next. Come on. Next. The estimated number of genes in human beings is about. Estimated number of genes. How many genes present? Not 80,000, not a 9, which is estimated. Option C. Okay, option C. Option C is answer. Estimated gene is a 30,000 genes. Okay, next question. Select the correct option that. Next. Select the correct option that correctly fill up the blank 1 to 4. 1 to 4. 1 to 4. 80. Okay. Okay, look here, guys. Less than dash genome represent the structural gene that code for protein. It's a NCRT line. It's a 2%. Less than 2% of gene. 5% no, 10% no, to 2%, not a 50%. Okay. The chemical substance that bind the repressor convert into non-DNA binding start is inducer. The structural inducer. Okay, the structural gene in a transcription unit is dash in eukaryotes, monocystonic in eukaryotes, and polycystonic in bacteria in prokaryotes, because NCRT lines. Option C is the answer. Option C is the answer. Ready? Next 89. 89, guys. 89. Which of the following? 89 question. Okay. Which of the following statement regarding human genome is incorrect? Human genome is incorrect. Okay. The human genome consists 3 into 10 to 9 base per about. Okay, correct. The average gene size 3000 base pair. Dystrophin is the largest in. Okay. The chromosome number contain maximum 2968 number of gene. Y chromosome that's least. Okay, correct. The repeated or repetitive DNA sequence are not. No, is a present. Option D is correct. It's incorrect. It's incorrect. Option D is an incorrect statements. Ready? Ready. And 19. DNA fingerprint. Arrange the various steps of. Next question. Arrange the various steps of. Various steps of DNA fingerprinting technique in the correct order. The first order, come on. Suppression of DNA fragment by electrophoresis. Digestion of DNA restriction in the nucleus. Hybridization using label deviant probe. Isolation of DNA. Detection of hybridized DNA fragment autoradiography. Transferring of DNA. Separated DNA fragments to nitrocellular membrane. Ready? Yeah. Which options is first? Isolation of DNA is the first. Okay, ready? Isolation of DNA. Two options are given. isolation of DNA. Okay. Then isolation of DNA, digestion of DNA, digestion, yes, digestion of DNA by restriction endonuclease. 2. Then next step is 1. Option A is correct answer according to our NCERT test book. You are ready? Option A is the correct answer. Option A is the correct answer. The first isolation of DNA and then digestion of DNA by restriction endonuclease. Then 1 is separation of DNA. Option A is the correct answer. Ready? Ready. The next question, matching the following, matching type of questions. So simple questions, guys, simple questions. Come on. Okay, ready? Which of the following pair is incorrectly matched? Incorrectly matched questions. Come on. It's a purine, adenine and guanine is correct. And pyrimidine, cytosine, uracil. Pyrimidine, yes, correct. Nucleosides, adenosine and deoxythymidine. Adenosine and deoxythymidine. Deoxythymidine. Yeah, it's a nucleotide. And DNA, basic biomolecule. DNA is a basic, no, not a basic molecule. It's a fun. No, DNA is not a basic molecule. Okay. Yeah, it's incorrect. But the old statement, purines, adenine and guanine, pyrimidines, cytosine, uracil, nucleoside, adenosine. Which option? Option D is answer. Okay, option D is the answer. Ready. The next question, match the column with the column 2 and select the correct option from the given codes. Okay, come on. Which is answer? So very important. Okay, the Frederick Misha. Which is answer? Frederick Misha. Yeah, nuclein. AL2. AL2. Oh, only one option AL2. Frederick Misha is nuclein. Next, Griffith, Bill3. Streptococcus pneumonia. Yes. Seal 4. Hershey and Chase, bacteriophage, yes. Deal 1, Watson and Crick, DNA double helix, and EEL 5, Wilkins and Franklin, X-ray diffraction data. Option A is the correct answer. As ready, option A is the correct answer. Ready, guys. The next, select the correct match of the enzyme which is related function. Okay, correct match of the enzyme with a related function. Okay, come on. So simple question. Enzymes. Okay, ready? DNA polymerase, synthesis of DNA strand. Yes, ready. RNA polymerase, synthesis of DNA. No, it's a synthesis of RNA. It's a third. Ligase, join short DNA fragmented together. Yes. So C is correct. Option D, B and C, yes. D, both A and C is correct answer. Option D is the correct answer. Simple question. The next, 94. Match the column with the column 2. Select the correct option from the given code. Okay. Come on. The sigma factor. Initiation factor. Yes. Yes, ready? The sigma factor, initiation factor. Come on, give me answer. Sigma factor is initiation factor. Which options? 2 options are given. A, B, A2. 2 options. B or C is answer. Then as capping is 5' end. 2 options are same. Capping is 5' end. The 2 options are C and 5. Tailing 3' end is correct. And then D, coding strand, deal 1 or deal 3, coding strand is, come on guys, coding strand is 5' to 3', deal 1, option B is the answer, simple question, okay, option D, okay, B is the answer, okay, ready, the next question, finish it, yes. Oh my goodness, super question. Yeah, related genetic code. Come on, which is answer? AUU is phenylalanine. Yeah, it's a fundamental. UUU is phenylalanine. 3 options are given, guys. Phenylalanine. Next, GGG. GGG is coded by. Come on, which option? It's a glycine. 2 options are given. glycine. The next is answer. UCU code Y, serine. It's a very, very important. Serine is the answer. It's a serine. Which is answer? Seal 1. Okay, option A is answer. Enough. Okay, so very important. You must study genetic code and their code by amino acids. Let's start. Select incorrectly matched pair. Select the incorrectly matched pair. Oh, select incorrectly matched pair. Okay. Initiative codon AUG. Okay. Stop codon uga, uag, okay. Methionine code by okay. Anticodons mRNA. Okay. mRNA contain only codon. Okay. The statements are incorrect. Which is answer? Option D is answer. D is incorrect. Okay. Answer option D is the answer. Ready? Okay. Ready. 97. Oh, 97. Come on, which is answer? 97. Come on. Okay, come on. Translation. Amino acyl tRNA synthesis. No. Which is answer? Amino acyl. No. DNA polymerase. Translation. DNA polymerase. Translation DNA polymerase. 1 and 2 selected option from the given code. Okay. RNA polymerase, no DNA. Translation amino acyl tRNA synthesis. Amino acetyl tRNA synthesis. Amino acetyl tRNA synthesis. Okay. The next transcription. RNA polymerase. Okay. RNA polymerase and DNA replication, DNA polymerase. So correct? Okay guys, correct? Yes, ready? I thought you ready. Ready. Okay. Which option? Option B is answer. AL1 translation, amino acetyl tRNA synthesis, and B3 transcription, RNA polymerase, transcription, DNA replication, DNA polymerase. Ready? Like option B is answer. Okay, next 98, question number 98. Match the column 1 with column 2 and select the correct option. Okay, ready? Okay, come on. Griffith, Lacapron, Jacob Monad. Yes, the answer is here. Oh my goodness, guys. A, B, C, D. Okay, A, B, C, D. ABC. Okay guys, come on. Which is answer? Griffith, Lacapron, semi-conservative DNA replication, transformation. Griffith, transformation, AL3. Which options are AL3? AL3. Two options are AL3. Two options. Griffith, Griffith. Which is answer? Griffith, transformation. Yes, AL3. Two options. Option A or B is answer. B. Next B. Jake Monad is Lacapron. Bill1. Okay, Bill1. Yes. AL3 B1 is option A is answer. Option A is the answer. Okay, ready. Next question. Match 99. Second last question. Okay. Column 1, column 2. Operator site behind the site of our. Okay. Last two questions. Okay, come on. Ready. Operator site. Binding site or repressor molecule. Yes. Binding site or RNA molecule. No. Binding site for repressor molecule is operator sites. Yes. AL2. Two options are AL2. Next. Promoter B1. Promoter site. Binding site for RNA polymerase. Promoter site. Okay. Regulatory gene and structural gene. Next, sealed regulatory gene. What is regulatory gene? Code for a protein enzyme. No. Code for a repressor molecule. Okay. So structural gene. Code for proteins and protein enzyme. Seal 4 and DL3. Option. Which option? Option B is the answer. Okay. Option B is the answer. It's a simple question. The last and final questions. Come on, give your answer. Give your answer, guys. Give your answer, guys. Come on. Yeah. Option B. Yes, option B is the answer. AL3, Alec Jeffrey, DNA fingerprint. BL2, Sanger, BL2, yeah, DNA segment. Seal 1, Jake Monad, Lacapron, yes. Deal for McLeod transform principle. Ready? It's good. Okay, guys, today finishing this section. Thank you for joining our NEET live. See you again next chapter, another chapter. See you another live. Okay, and bye bye, guys.